Hello, everyone. Welcome back to our event. We're starting now the afternoon part of this day. And the first lecture that we are going to have today is, actually, not today, but now, is Dr. Lisa Baumann. So welcome, Dr. Lisa Baumann. Thank you very much for accepting our invitation to be here today. Dr. Lisa Baumann is a researcher and a group leader at the University of Heidelberg, and her team is currently doing research in the field of endocrine disruptors, aiming to optimize international guidelines regarding this matter and to close knowledge gaps for the endocrine regulation of vertebrate developments. And she's here today to talk a bit with us about using zebrafish as a model for thyroid hormone system disruption. And we also would like to invite you all to post your questions here in the chat, and be asked when the speech finishes. Thank you again, Dr. Lisa.
Thank you very much for having me. I'm very happy to be here and to be part of this great webinar series. It's a great effort that you're making, bringing all these researchers together, working on different topics with this great research model. And I would like to talk about our work in different projects, the Ergo project and the IFED project. That is a very applied aspect that maybe some of you are not so familiar with. So it's the field of ecotoxicology. We work on pollution of aquatic systems and use the zebrafish as a model for that. The slides I'm showing you today is a big effort of many people. You see some of the co-authors listed here, the big projects working on this. I invite you to visit our webpage, to follow us on Twitter, to see what we are doing to improve research on endocrine disruptors and establish the zebrafish as the model to investigate these compounds. So let's see if this works. Yes, it works. So as I know that the audience here is very diverse, I start with the definition. So what is an endocrine disruptor? There is a definition by the World Health Organization that says that endocrine disruptors are exogenous substances or mixtures that alter functions of the endocrine, so the hormone system, and consequently, this is the important part here, cause adverse health effects in the organisms or subpopulations. So there has to be a negative effect, for example, on reproduction of animals. There's many famous examples for that. Many of you know, for example, that contraceptive pills end up in the water, feminize fish. But there's other famous examples listed here, some pesticides, bisphenol A, all of you know the plastic problem. Not only the plastic itself is a problem, but the softeners that are used for them, all of this ends up in the water and can have an influence on fish and other organisms. And we use the zebrafish to investigate this. For the following slides, it's very important to understand that environmental pollutants act on different biological levels. So all of you are scientists and know how biology works. We start at the molecular level, then go up to cells, organs, the organism, the population, and then the ecosystem. And environmental pollutants can act on all of these levels, but with a different velocity and sensitivity of the reactions. So the lower we go in the organization at the molecular level and cellular level, those reactions are really fast and sensitive. But the ecological relevance is at higher level. So then we talk about populations and ecosystems. So for example, if an entire fish population feminizes, this has, of course, impact on survival of the population. So this is something that you should have in mind here, that we talk about all of these levels, and this is really relevant in ecotoxicology. And this relates to the adverse outcome pathway concept that was developed a few years ago that takes this into consideration, so that environmental pollutant starts interacting with an organism at molecular level. So at the left side, for example, binds to a hormone receptor. Then we have a cellular response that translates into an organ response, an organ system response, and then the organism and population. And this is where we see the adverse outcome. And okay, now I see my slide animations don't work Yeah, that's not important. There was an example here showing how a compound that interacts with the estrogen receptor leads to changes of a certain protein, then changes in the gonads, feminization of the organism, and then the population and decline in reproduction. So this is the famous example that we know for many years. But estrogens are not the only important hormones that we talk about. So this is a very simplified picture here for you for the different endocrine systems we have in vertebrates. And as you know, zebrafish is a vertebrate. And these endocrine systems are very conserved. So the zebrafish estrogen receptor is exactly the same as in humans, and the same for the thyroid receptor. And in ecotoxicology, we are mainly interested in two targets of endocrine disruptors. So one is the gonads that results in reproductive disorders, and another one that is coming more and more into focus is the thyroid hormone system. If we disrupt that system, there can be severe developmental and physiological disorders. That we observe in humans and in animals. And this is a serious environmental and societal problem. There's different estimations, but probably over 1,400 compounds are in the environment that affect humans and animals and impact the hormone system. So, coming to our fish, the zebrafish. So how can we investigate thyroid-related effects in fish? Fish have a thyroid system. They have thyroid follicles, just like in humans. We can find it in the jaw region next to the gills. There's the thyroid follicles where the thyroid hormones are produced. And these hormones regulate different processes, especially during development. So we know that, for example, fin development and skin pigmentation are regulated by thyroid hormones, but also very important organs that are important for survival, like the swim bladder or the eyes, are regulated by thyroid hormones. So if we investigate the development of these organs under exposure to compounds that disrupt the thyroid hormone system, we can estimate the effect on populations. We also have to, of course, investigate thyroid hormone levels or gene expression of thyroid-related genes in the target systems to make sure that what we see is really specific for the thyroid. So the ones that are marked here in red are the ones that we are focusing on at the moment in our different projects to see how we can investigate this with our zebrafish model. I will start from the molecular level up to the population relevant level. So first thing that, or not first thing, but one of the things we're currently doing is measure thyroid hormones in fish. And this may sound very simple, but I was told it's not. Luckily, I'm not the one doing these measurements. Rikke Poulsen, that you can see here from Aarhus University, did a fantastic job in our Ergo project to measure thyroid hormones with HPLC in zebrafish embryos, and not only in the entire animal, but also in different tissues. So, for example, the right figure that you can see here is thyroid hormone levels in the eyes. So we dissected the eyes of 5-day-old zebrafish and measured how the levels change with exposure to different compounds. You can see the white bar is the control, and then we use PTU or TBBPA. Both are thyroid disrupting chemicals, and you can see that PTU decreased the thyroid hormone level in the eyes, and you will soon see that this also makes a difference for the eye development. This is a very challenging and difficult method because, as you know, it's not that easy to get a lot of blood from our small fish, but it is possible. This will hopefully soon be published. It's currently under review. Then you can read more about this. Another thing we are doing extensively is histopathology is very important. And this is our PhD student, Lisa, who did a great job with all the histology analysis. And this is from another experiment with PTU, where we exposed the animals a bit longer for 63 days. And we investigated the thyroid and other endpoints in those fish. And you can see the PTU treated ones. They really show, you can really see the thyroid follicles. They are proliferating, they get bigger. So PTU inhibits the thyroid hormone synthesis. And what's what's happening is that the organ tries to compensate this and gets bigger. And we can see this in the histology, measure the follicles, and then make those relationships that you can see in the figure. So the more PTU we give the fish, the bigger this thyroid follicle gets and the more active this organ gets. So this is a very sensitive endpoint that we can measure in fish to see if a compound disrupts the thyroid hormone system. Okay, so this is not working. I'm sorry, I can try to click, but no, this will not work. Okay, this is really a pity. I'm sorry, I was not familiar with this system here, how to share the slides. I had to upload them. So there were 2 videos, but I'm sure you can all imagine this. So we are using zebrafish embryos, a transgenic line that was kindly provided by the Costaiola lab in Brussels to us, and they express fluorescent signal in the thyroid follicles, thyroglobulin. And if we treat the animals with thyroid disruptors, in this case, again, PTU, those follicles get much bigger. So you can already see it in a 5-day-old zebrafish embryo, how a compound disrupts the thyroid hormone system and how the organ itself reacts to it. So this is a very simple way to do some imaging analysis. This can, of course, all be automated. You all know this better than me. And then we can simply measure how big are the follicles, how high is the fluorescent signal, and then relate this to the compound we exposed the fish to. Then the other thing we're looking at, as I mentioned earlier, is the eye development. So we know from all vertebrates that thyroid hormones are really important for proper development of the eye. All the different layers in the retina, the photoreceptors, the pigment epithelium are dependent on thyroid hormone signaling. So what we do is histopathology of zebrafish embryos where we focus on the eyes. You can see here a picture from a 5-day-old embryo with the different layers. And just for those who are interested in the technique, this was not developed by us, from a different lab a few years ago. We embed the tiny embryos in agarose and then just cut the entire agarose block with the embryos inside, multiple ones, so then we can really quickly analyze different animals. And then we measure the different layers of the retina to see if there's changes due to the treatment we did. This is some examples here. For example, we measure this pigment epithelium, this dark layer at the back of the retina. And you can see that with PTU exposure, this layer decreases in diameter. And also with perchlorate, this is another thyroid disruptor. And you can really see it on the picture. Also, the pigmentation gets lost. The entire structure of the eye is changing. I forgot to say one thing. So the PTU we are using is not the one that many of you certainly use in the lab to bleach the embryos. Unfortunately, there's 2 different PTUs. Phenylthiourea is the one used for bleaching, and we use propylthiouracil. Horrible word. I'm sorry for my pronunciation. And this is one that specifically targets the hormone system, but it has the same effect on pigmentation. So you can really see that the eye looks very different in those animals, and this is certainly problematic. I mean, the optic sense is very, very important for survival. We also see malformations, not only changes in the different layers, but you can see here that this inner plexiform layer in animals treated with iopanoic acid shows these malformations. So normally in the figure below, you can see that this layer should be nicely delineed around the retina. And in the upper one, you can see that, the structure is really changed. And this can also have an effect on the functioning of the eye, of course. We also do some analysis of the photoreceptors. With different methods, we have transgenic models or antibody stainings where we look at the different photoreceptor types and how the composition changes in the different treatments. So we know that this the ratio of different photoreceptors is very important, and we can see changes with our treatments. in the composition and also the size of the photoreceptors. And it's likely to assume that this also has an impact on the function of these structures. So this brings me back to the AOP concept. If you remember the beginning of my presentation, that we always want to look from the molecular level up to the population relevant level. What's happening if we treat our animals with different thyroid disruptors. So in our case, we have already shown that thyroid hormone levels change with the treatment. We see that the retinal layers change in the treated animals, so that there's structural changes. And now we were interested to see how this translates into behavior and then maybe mortality on the population level. What you can see here was just recently published. You can find the links below. This is an AOP concept, a combination of a literature review and an AOP that you can find online. And this contains really a lot of data. And we did this for a very simple reason, COVID-19. Our PhD student started her lab, her work just before the lockdown. And then we said, well, then it's literature work. And she assembled all the literature and then later combined it with our data. And we have this really nice AOP now showing the entire chain from thyroid hormone level changes up to mortality that is relevant for the population. So this is very, very important also from a regulatory perspective, because this shows that if, for example, a pesticide enters a river and then attacks the thyroid hormone system, this will have an impact on the survival of the fish. And by that, maybe it will be banned one day. There's also other AOPs that are already established. I want to encourage you to read those. You can see the numbers on the right and the authors. So Dries and Lucia from Antwerp, University of Antwerp, are working on this since many years, also in our Ergo project, and they nicely showed the entire chain from altered thyroid hormone levels to impaired swim bladder inflation. You can see an example below on the left. So even in the very young zebrafish stages, you can see that the swim bladder does not inflate properly if we disrupt the thyroid hormone system. And also in older life stages, we only see one of the two chambers. And this has, of course, an impact on the swimming performance of those fish. And very self-evident, a fish that cannot properly swim has a problem with survival, and the same with a fish that cannot properly see. So these AOPs are very relevant to understand effects that we observe in the environment or that could potentially be observed if some chemicals are released into the environment. So I encourage you to read the wiki. Really, all of you that don't know this is really a nice collection of work where you can find literature, correlations between different effects. And we need more people to work on this and to put their data into the AOP wiki. Okay, so this is also not working. I'm really sorry for that. But I'm sure most of you are familiar with this. What I wanted to show here was a plate of zebrafish embryos, a 24-well plate that was exposed to different light levels and colors, and we were tracking the movement, with these typical behavior boxes that probably most of you have. And then we can see changes in the swimming behavior caused by the chemical treatment because the eyes are not well developed or the swim bladder. And this is just one example here of what's happening. Again, the control fish. So they show higher swimming behavior in the dark and lower in the light. And this is really a dramatic change that we observe here. And another thing that I need to mention here is that, of course, we are in concentration ranges of those compounds that are not toxic per se. So endocrine disruption is not toxicity. So it's not like those animals are close to dying. Not at all. They look more or less normal and swim normal. But if you measure it, you can see that they are not normal. So this brings me to this applied aspect, why we are using the zebrafish and why this is important. So if a company, a chemical company, wants to sell a product in Europe, in the US, they have to test the compounds for toxicity, but also for endocrine disruption. And there's different guidelines for that from the OECD using different animals and to investigate different endocrine modalities. So there's different endocrine axes, estrogens, androgens, the thyroid, steroidogenesis, and there's different tests for that. And you can see that also amphibians are used, so the xenopus model, and then the fish for many other tests, but the thyroid, is only tested with amphibians. And the reason is very simple, because amphibians undergo metamorphosis from tadpole to frog that is regulated by thyroid hormones. And if you expose them to chemicals, this metamorphosis is disrupted, and you can easily see this. In fish, it's a bit difficult to see. So there's no thyroid tests for fish, no established test that is validated by OECD. And that's very important to have such tests because fish are the main test organism. The amphibians are only used for the thyroid. And we could avoid a lot of animal tests if we could assess everything in one model in one fish, for example, the zebrafish. So that's what we're really specifically working on. So not only to understand how the thyroid hormones regulate development of the eyes or the swim bladder, but also how we can test this with the OECD tests. So what we have done so far in Ergo and the IFAD project is that we tested those 3 guidelines that you can see on the bottom left. These are the numbers of the guidelines. The 236 is this typical normal 5-day zebrafish embryo toxicity test, where, for example, toxic levels of compounds are assessed. Then there's the 210. This is for developmental toxicity. And the 234 is for sexual development. So this is already a test for endocrine disruption, but it does not cover the thyroid hormones. And in the table, you can see which thyroid-related endpoints can be assessed in which guideline. And you see a lot of green checks there. So actually the thyroid hormone levels can be measured in any test. The thyroid follicles develop already at day 3. We can measure them in all of these tests. The eyes are one of the first organs to develop because they're very important. The swim bladder too, the anterior part, so the second chamber, develops later, so this cannot be assessed in the embryo, but the posterior chamber. And then the swimming behavior can, of course, also be assessed in older life stages, but we have not done it yet. So, but at least in the embryo, it works really well to assess thyroid disruption. So we are on a good way. We are in contact with all the authorities from OECD, from the EU. They are also funding us for developing those tests and implementing them into the OECD framework to be able to assess this in fish very soon. We need to go into validation. This will still be some work to confirm that what we do can also be done in other labs. So if any of you is interested in that, feel free to contact me. There's still a lot of work to do, and it's very important work, I think, because it really has this applied aspect to protect the environment from pollution. And I will show you the other project, the IFAD project, to really make clear how this is important in this context, because there is already existing guidelines, and it's much easier to improve them than to develop new ones. So we had 2 hypotheses for this project. The first one was that it's possible to merge 2 of these guidelines without making any changes to the protocol, and then that we can implement new endpoints in these guidelines. And by this, we hope to speed up the process. So normally it takes many years at OECD level to validate such methods, but by merging existing guidelines and only adding new points, we hope that it will go faster. And then the approach was to run this new protocol that we developed with 2 different well-established endocrine disruptors, one for the thyroid axis and one for the estrogen axis, to also show the specificity of the effects. So this is the test that we developed or that we changed. So there's the two guidelines, the 229 and the 234. The 229 uses adult zebrafish. And this is a typical reproduction assay. So adult fish are exposed for 3 weeks. And then every day the eggs are collected and counted and checked for fecundity. Then there's also gonad histopathology. Vitellogenin is a very important biomarker for estrogenic effects. And that's it. So this mainly checks for the effects on the estrogen and androgen axis. But we suggest that we can also add hormone levels, the thyroid follicle histopathology in this first test to also check for thyroid effects. And then the second test, the FSDT, the fish sexual development test, takes 60 days. So it starts with fertilized eggs until the late juvenile stage when the gonads are differentiated, and again has the strong focus on the estrogen and androgen axis. So growth, hatch, survival, vitellogenin, the sex ratio, and the gonad histology are assessed. You all know that zebrafish can easily change their sex if we expose them to chemicals. So this is very, very useful for assessment of endocrine disruptors. But as you already saw before, we can also use it to assess thyroid endpoints. So again, we suggest to check hormone levels, thyroid follicle histology, the eye development, and swim bladder inflation, and to combine those two tests. So we have the adults, and then we use the F1 generation of those. So then we have a 2-generation test that covers all life stages and all modalities in a comparably short time frame. There's other tests that take much longer. Zebrafish develops really fast. This is a very high advantage. By that we can quickly assess this with the zebrafish model. So I will just show you a few examples of this. So the first phase where we exposed the adults is when we assess the reproduction. You can see on the left PTU, the thyroid disruptor, and on the right EE2, that's ethinyl estradiol. This is a very, very strong estrogen. This is one of the main components of the contraceptive pill. So many, many women all over the world take this every day and put it in the toilet. And this ends up in the river. And you can see the concentrations. So 10 nanogram per liter is already enough to stop zebrafish from breeding. They are not breeding anymore at all. You can see this lower in the figure. So after 10 days, the fish were not breeding anymore. And these are actually concentrations that we find in rivers all over the world. PTU also has an impact on the reproduction, but they were still breeding at the end, even though the concentrations were really high. So reproduction can be disrupted with estrogenic and thyroid effects. We know that the hormone systems interact with each other. They are not isolated. And this is a nice example for that. But then we have very specific endpoints, biomarkers, vitellogenin. I don't have time to explain all of this. I hope some of you are familiar. So vitellogenin is the precursor protein for the egg yolk to feed the embryo in the egg. And this is controlled by estrogens. So if we treat a fish with estrogens, this will increase in concentration. And you can see this in the left figures with the males. So males normally don't produce much of this protein, but if we give them ethinyl estradiol, it increases. And it does not with PTO, which was a really nice confirmation that our biomarker is specific for estrogenic effects. It's not only important to understand if the thyroid, if the hormone system is disrupted, but we would also like to know the mechanism behind it, of course. You already saw this slide. This was from the IFED project, which is why I show it again very quickly. So these are the F1 generation from the parents that were exposed. In the parents, we saw changes in the thyroid histopathology, but also in the F1 generation, and it was really strong. And we did not see this with E2. Then we checked for the eye histopathology, and you can see that PTU and ethinyl estradiol had effects because, again, we have an interaction of the endocrine systems, and also estrogens are a bit important to regulate the eye development or overall development. So this is sensitive to both mechanisms. And we also found that early life stages, the younger fish, are more sensitive to it, which makes sense. The eye develops really fast in the first days because it's really important for survival. And if we disrupt it in that phase, we can really see changes later. There's some recovery that we observe, and then the effects are not that strong anymore. But still, we think that if the eye development is disrupted so early and so strongly, that this will have effect on survival of the early life stages. So this brings me back again to our AOP. So again, in this experimental setup, we could show from the molecular up to the morphological level that disruption of the thyroid hormone system has a strong impact on the eye development, and that this will certainly result in mortality. Again, I encourage you to read this article because we have listed all the other studies. There's great research ongoing all over the world. There's studies recently showing that they did predator avoidance experiments showing that fish treated with thyroid disruptors have developmental changes in the eyes and cannot recognize predator fish anymore. And this means, of course, that they will die. So this is really, really relevant. Oh my God, this does not look well. Okay. Due to technical reasons, I'm really sorry for that. I have to skip that. It's actually just a summary of the results from the experiment showing that, both compounds had specific effects on specific endpoints and that we can implement them in the existing guidelines and to show effects on all the different endpoints for the different endocrine systems. I have to mention the funding here, really important. It's really great seeing over the last years that there's funding provided for such applied research. It's really important to develop those guidelines and to work on this, even though some parts may scientifically be not so exciting, but this job needs to be done. And many great people support us in this, in the IFED project and also the Ergo project. And I would really like to say thank you to everyone who is involved and who provides funding. This is my last slide to refer to the Ergo project. So feel free to contact me under this email address. I forgot my Twitter account. I'm also on Twitter. I'm sure you will find me. And the Ergo project, where we regularly post what we are doing with all the great partners. And with this, I would like to thank for your attention, and I'm happy to receive questions. Thank you.
Hello. Thank you very much for this marvelous presentation. I think we are all happy. There is only one question for you right now.
It's probably in the comments, not in the private chat. Yeah, I can see it.
Yeah, yes. It's the question is, in the future, will it be possible to test mixture of substances with these protocols or some new protocol?
Yes, this is a very important and good question. This is really something we are aiming for, and we are currently working on project proposals to do exactly that. And this is on different levels. We know that mixtures are the thing we have to test because this is what we find in the environment. Of course, there's not only one chemical, there's hundreds or thousands. So we have to test mixtures, and this is something we're doing. And already many people did. There is many projects ongoing with this. It will be a bit difficult to put this into a regulatory context. What is a relevant mixture? How much of which compound? This is something we have to debate. But of course, we can go out in the rivers, in the sea, measure what's in there, and then use it in the lab and see what happens. So this is of course something we need to do more. Yes. Okay.
The next one is from Bianca, and she's from the University of São Paulo. In the AOP, do the concentration of the chemical alters the possible outcomes?
Yes and no. Of course, the dose makes the poison. This is a very established fact. So the more you use, the higher the effect will be. But as I mentioned during my presentation, for endocrine effects, it's very important to have in mind that this is not unspecific toxicity. So those animals are far from dying. They are not in this. These are not lethal concentrations. So the maximum mortality we accept in those tests is 10%. So what, and then we have the low X and no X, so the lowest observed effect concentrations and no observed effect concentrations, and you can make your AOP with different concentrations, of course. What the only thing we have to ensure is that we talk about sublethal effects. But of course, if you give less of a compound, then maybe you will see an effect at the molecular level. So, for example, activation of the estrogen receptor or VTG induction is something that reacts really fast. But if the effect is not very strong, it will not translate, for example, in impaired reproduction. So this is also a very theoretical thing, the AOP, what could happen. This does not mean that it does necessarily happen. It can also stop at a certain point. So, yeah.
Okay. The last question is from Gisele. She's saying, congratulations, great work. Thyroid hormones alters growth factors like GEH, IGF-1. And she would like to know if you see zebrafish growth alterations in size, weight, and metabolism.
Yes, we see very strong changes, and exactly for that reason. Thanks for that comment. Thyroid hormones are super important for growth. I didn't have time to show all the data, but the fish are always smaller. In most of the cases, they're really smaller, they weigh less, even though we are not in toxic concentration ranges again. And we are, of course, also interested in the metabolism. So this is ongoing work. We see that the liver metabolism changes. This has, of course, also effects on detoxification and on nutrition status. This correlation is very strong and really important. And we also know this from cohort studies with humans. So children that were exposed during pregnancy and during the early childhood to thyroid disruptors are often smaller than normal children. And the other very, very important thing is the effect on neurodevelopment. So we also see decreases in IQ all over the world that is related to chemical exposure and could be a direct effect of thyroid hormone disruption.
Okay. So thank you very, very much. Renata is also thanking you for this great talk.
I thank you very much for inviting me. It was really a pleasure, and I hope you enjoyed it too. And I look forward to more presentations. And thank you for the interesting questions. And feel free to contact me if there's any more questions or you want to collaborate with this. Would be really nice. Thank you.
Okay. Thank you very much. And we're going now with our sponsors video, and later we'll be back with another presentation. See you later.
My name is Mark Francis. I'm the founder of Aquaneering. For the last 20 years, Aquaneering has built a team of dedicated professionals to support your research. Our mission is simple: provide the highest performance zebrafish housing at a reasonable cost. Our team is here to allow you to focus on your research.
We at Alesco work daily to create solutions and equipment for the scientific community, contributing to the advancement of Latin America biomedical research. Our mission is greater than just providing equipment. It is to provide security with a quality service and mainly with enormous respect and attention. For this reason, we invest in high technology and seek to maintain long-lasting relationships. Our commitment is to be a reliable partner that understands the needs, the reality, and the conditions of each customer to always offer the best solution. We understand the benefits of scientific research for humanity, and this encourages us. If today we have a better quality of life, greater longevity, if we beat cancer or use a medicine for a headache, it is because the advance of biomedical research allows us to. We trust in the work of researchers, in science, and the scientific community, and we pride ourselves on doing our part. Just like you, we are passionate. Science is what moves us, because for Alesco, research is for life.
Welcome everyone to FST's presentation. We appreciate you taking the time today and look forward to helping you find the right tools for the right application and your endless discoveries and research. Find Science Tools has been in business for over 45 years, providing scientists, researchers, and other life science professionals with high-quality surgical instruments utilizing German steel and German craftsmanship with a secondary quality control inspection in our Heidelberg office. As we say, we only carry the best and offer a multitude of fine microsurgical instruments like spring scissors, forceps and rongeurs, but also a variety of options for wound closures and animal identification. In addition to our 3 FST offices, we have over 50 distributors worldwide. While the majority of the audience is based in the USA, please note that we can ship anywhere in the world, and any products not listed in our physical catalog or online at findscience.com can be sourced as a special order, and we will work with you to get you what you need. Before we start, for those who are unfamiliar with FST, let me go over our core values we hold ourselves to and strive to provide to our customers. Quality. Impeccable product quality is what differentiates FST from the others. Secondly, our customer support. We strive for 100% customer satisfaction. Lastly, our QC department in Germany upholds our manufacturers to the standards FST and our customers expect with every instrument.
Saúde, direito de todos e dever do Estado. Direito à moradia, ao emprego, à educação, a um meio ambiente saudável, à água potável, ao lazer, à cultura. Direito à voz e à paz. Respeito à diversidade. Saúde para a Fundação Oswaldo Cruz abriga todas essas dimensões. É assim desde 1900. Uma visão integrada da saúde. Na pesquisa, ensino, assistência, informação, comunicação, memória, na promoção da saúde, na inovação, onde permanentemente se pensam respostas para as necessidades da sociedade, onde se faz ciência em defesa da vida. Você pode até não saber, mas carrega a Fiocruz dentro de você. Desenvolvemos e fabricamos vacinas, medicamentos, biofármacos, testes para diagnóstico. Formamos pessoas do nível médio à pós-graduação. Realizamos pesquisas para superar ameaças à saúde, para diminuir riscos ambientais, para prevenir doenças e agravos. Pesquisas que beneficiam crianças, adultos, idosos. Uma instituição pública e estratégica de Estado, integrante do Sistema Único de Saúde, com uma rica história de contribuições à sociedade. Presente de ponta a ponta no Brasil, onde cada trabalhador é um elo forte e ativo. Nela, ciência e saúde cumprem uma função social para o país e o mundo. Pés fincados na tradição, olhos voltados para o futuro. Somos patrimônio da ciência e da saúde, da humanidade, do povo brasileiro.
Generate and spread knowledge to form transforming agents. This is the goal of the biggest university of Rio Grande do Sul, State Northern Region, and one of the biggest from the country's southern region. With half a century of history, UPF maintains its roots in educational excellence, research and extension incentive, and the constant search for technological innovation. There are 9 campuses that are distributed around the region, and 12 academic units, offering undergraduate, specialization, master's, doctorate, and postdoctorate courses. The infrastructure is a great differentiator. There are clinics, rooms for practical experimental teaching, laboratories, amphitheaters, auditoriums, and libraries with hundreds of thousands of titles available, both in physical and digital format. UPF stands out for the quality of teaching, academic, investigative in nature, the dialogue with the community, the performance in the job market of professionals who have graduated from this institution, and by the qualified teaching staff with teachers who promote the renewal of methodologies for the teaching and learning process. The research incentive in the university generates and disseminates knowledge. The extension reinforces the university's commitment to the community, with projects and programs that, through services provided, approximate academics to social reality and contribute to the sustainable development. Along with a university social responsibility policy, the institution promotes citizenship, environmental awareness, social inclusion, culture, and development. Major events are held at the university. An example is the National Journey of Literature, one of the greatest literary manifestations in Latin America. The Radio UPF and UPF TV broadcasters with open signal and educational character provide the community with cultural programming focused on local development. Facilitating access to higher education, the university has several lines of credit, scholarships and funding, benefiting more than 70% of students. One of them is the PI, UPF's own financing program. Every semester, possibilities of exchange are offered to students through agreements with institutions in several countries. Acknowledgements strengthen the greatness of UPF. The university is among the best private institutions in Brazil. Its courses received grades 4 and 5 in the assessments made by the Ministry of Education. The institution has also received in recent years other important awards, such as Social Responsibility, granted by the State Legislative Assembly. The Planalto Medio UPF Scientific Park contributes directly to the development of the region. Connecting innovation, science, and technology, the UPF Parque, with its technology-based incubator, supports and encourages innovative projects and local entrepreneurship. Providing quality of life, UPF offers to the academic community services that favor inclusion and permanence in higher education and the construction of great stories. The university has a community center, a leisure environment to eat, interact, and rest. This is the University of Passo Fundo, an institution that prioritizes ethical and humanistic formation. UPF, knowledge is our nature.
Hello, everyone. Welcome back to our next session. And for this one, I'd like to welcome Dr. Marie-Laure Bergot. Thank you very much for accepting our invitation to be here today, speaking with us. Dr. Marie-Laure is a researcher at the Marbeck since 2002, where she conducts research on fish adaptation to environmental challenges. Her research interests are either with an applied focus, such as fish welfare, and or an environmental perspective, such as understanding coping ability and contributing to modeling fish individual and population traits, including life history traits. She's here today to talk with us using zebrafish in ecotoxicology in a behavioral perspective. We'd like to invite you all to post your questions at the comment section, and they'll be asked when her speech finishes. So, welcome, Dr. Marie-Laure. Thank you for being here, and you can start your presentation now.
Okay, thank you very much. I'm glad I was invited to give this talk, so I will try to share my screen. Hopefully, everything will work nicely.
Yes, I'm sure it will.
Okay, so, oops. Okay, so here we go. You have my presentation on your screen.
Yes, it's working. You can click on hide.
Oh, yes. Good. Okay. Well, so I will present this presentation, zebrafish use in ecotoxicology, behavioral perspective. And this is a presentation that we built together with Xavier Cousin, a close colleague of mine. And we are working in two different institutes. I'm working for Ifremer and he's working at INRAE. And we are actually located in the south of France in Ifremer Marbeck laboratory. So we have been developing quite a lot of work on using zebrafish. And I have built my presentation around first some definition and concepts, and then I will illustrate with different work we have been conducting. So concerning definition, I think it's always quite, for me, it was when I started this field of toxicology, I found it was sometimes complicated to understand what was what exactly. So I went to the basics. So I will try to give you some definitions. And then I will put the behavior value in context, in the context of ecotoxicology. Zebrafish, of course. Zebrafish for its behavioral components that we can use for ecotoxicology purpose. And then, yeah, some illustration. We have been mostly working in ecotoxicology using organic contaminant, so either PAH or PCB, PBDEs, we all are organic persistent contaminants. And we have been using zebrafish for this. And more recently, we also have been working with microplastics and other compounds. But yeah, this is the focus of today. So concerning, as a start, definition. So what is important to imagine that it's when there is a toxicant exposure, it can be through many routes, as you all know. It can be via the air, via the water, and of course here we are more going to talk about aquatic animals, fish in the case. So it will be mostly contaminants that are found in the water compartment, but it can come from the atmosphere, or it can come from runoff from the lands or from the rain. So the sources are really very complex and multiple. But let's say the toxicant is present, it is entering the body of the fish either by ingestion, because the prey is contaminated, by the skin, or by inhalation in case of terrestrial animals, or via the gills for a fish. So the toxicant will be absorbed, will diffuse through the bloodstream, and be distributed into the body, the different tissues, the different organs. And speaking about organic contaminants, they will mostly locate in fatty organs. So because they are largely lipophilic, so they will combine with lipids, and they will eventually be stored in the lipid compartment, and they will eventually already generate some toxicity, and then the body will react to this. For many contaminants, by chance, they are metabolized. What does it mean? It means that the original compound will be changed. Most of the time, it is degraded in something that is less toxic, metabolites that are often hydrophilic, so they will be excreted, so the animal will get rid of it. But sometimes the metabolites themselves are even more toxic than the original compound, and they can be reabsorbed and stay in the body of the animal. So this is the main picture. What is also happening is that the metabolite, but also the toxicants that are in the storage compartment can be excreted, and one of the important way is via egg production. So this is a very important way of excreting toxicant, and then you will have either toxicant or metabolite, and they will go back to the system. So it's a loop that can be present for a very long period of time, especially with some chemicals that are very remnant, very persistent. So this is really for toxicology. So people working in toxicology, they will explore the different routes, the different exposure modes, and just focus on this story. Now, if we go from toxicology to ecotoxicology, what is added is mostly the chemistry and the ecology. What is, surrounding the animal and how this can interact with the contaminant and the way the contaminant is acting on the animal. So what for? And this particular field of ecotoxicology is mostly used for, has been developed at the base for managerial aspects, because once you integrate toxicology, chemistry, ecology into this ecotoxicology, what you can do is risk assessment. So you can evaluate the problems posed to the ecosystem, to the humans and other living beings. And you can eventually try to manage the environment. So really the important thing here is that you can try to, act and improve the situation. Of course, it's not only chemistry and ecology, although this is already complex in itself, but you also have other influences that will come in the story, because you will have the physiology, biochemistry, limnology, genetics. All this will make the system more or less sensitive to the original toxicants that has entered the system. And of course, the environment is also something very complex with different meteorology, oceanography. So you can have a lot of different aspects. And all this will actually help you to distinguish the major influences on your system. So this is really how ecotoxicology was defined at the start. And it's not really that long ago. It's about only 30 years ago, 20 years ago, sorry, that ecotoxicology was really defined as a field of expertise. And how can we use it? How do we understand it in terms of levels of investigation? Because when speaking about animal biology or ecology, we can think about an environmental stressor. We have all heard about climate change and other stressors that are currently applied very heavily on our ecosystem. So consider any environmental stressor, and then there is a mistake. And then you can consider different levels. You can either think about sub-organism scale, individual scale, or population scale. You can decide which scale you want to work out. And then the other very important scale is the time, because maybe you want to investigate things that are happening within minutes, days, months, or years. And obviously, you will not be able to do all these different fields of research at the same level. So this is to put in perspective the different things that you can do and where it's actually relevant. Like, for example, bioenergetics will be something that will be focused mostly on the individual, and it will be in weeks or months. So yeah, as an example. And the more you go to the right and to the population, you increase your ecological relevance, because this will be the output of the research will be more useful for risk assessment or environmental management, for example. At the opposite, you go through detailed research and you can tackle mechanisms behind the defects that you observe. Okay, so if we put again this different definition, so you can imagine your stressor is a toxicant, and here at the individual level, you will find again organs, metabolism, function, like I just showed you before. So here we are, here we are mostly in the toxicology, like we said, environmental toxicology and behavioral toxicology. So it's really a world in itself, because you have all these different possibilities. And the more you go for the population level, then you go more into this ecotox, and even behavioral ecotoxicology, which is becoming quite a new field of research. Of course, in this case, you can use field approach. It's more top-down, it's descriptive, analytic, and integrative. Whereas in the toxicology, like I just said, you can do bottom-up research, dose response effect, cause effect, and be mechanistic. So this is really to give you this definition, and now we will be focusing more on behavioral ecotoxicology. And this is another, just I give you this so that you have this reference of Hinton 2005. I was really very happy the day I found this paper, because it was really explaining in details why you have to choose among these different levels from molecular to community. And this is very helpful when you need to argument why you are doing your research at individual level and not focusing on molecular. Or it's actually really this question of the link between mechanistic and ecological relevance on one side and the other side. So I invite you to go into this, and it's very helpful to write an introduction to a master report or something like this. It's really full of ideas. And myself being really focused on behavior, of course, it's at the individual level, and it's very important because it's exactly at the mid-level between ecological relevance and mechanistic understanding. So now we go to the next step, which is what is the behavior value in ecotoxicology in particular. So this is quite an old drawing I have remade from a paper, really old itself, but I find it very clear and very easy to put in perspective what does a perturbation at some time to an animal and its population. What you have to understand is that when there is something happening, like a pollutant, the first response will be very fast and will be at the individual level, because you will have a behavioral response of escape, for example, and then you will have some physiological homeostasis going on to help to acclimate or to adapt. You can have this physiological complete response, and this is still individual response. But there is a point in time or a point in scale where you will have some differential deaths and eventually a change in death rate. And then, of course, you will change your population in the end, because if all the animals sensitive to a particular, heat wave are dying and not the other one, you're changing the composition of your population. Eventually, you will change mortality and fecundity of a particular species. And this will, of course, as a consequence, change the genetic compartment of your population or of your ecosystem composition. And this go to a selection process. So here we are going from the individual to the population response. So it's very important to have this in mind, because when we are working in behavioral ecotoxicology, of course, we are focusing on these very first levels, and we can detail them further. So we can have this view of Scott and Sloman. They have really been focusing on the effect of pollutant on complex fish behavior. And the idea, their main point is that you have a stimulus, whatever it is. Sensory perception will be the first level of interaction between the pollutant and the animal. There is some central processing going on. And most of the time, what will happen is you will have, or depending if you're speaking to a physiologist or to a behavioralist, you have either behavioral responses coming first, like escaping or strong avoidance, or you will have, and at the same time physiological responses. So it can be most of the time everything is happening quite simultaneously, but yeah, you have these hormonal changes, cardiorespiratory change. When the fish is escaping from a threat, it will have an increased heartbeat, for example, and then a cascade of hormonal changes. So what Scott and Sloman have better defined is all the different values. So if you need to present an argument about why you are studying this particular behavior in your work, then this is really an interesting classification. So what they first distinguished was the behavioral effects of aquatic pollutants, and the three main that they put first was predator avoidance, reproductive behavior, and then social behavior. Because we have all this response to the chemical that are very intense in fish, because fish are extremely sensitive to smell, to different molecules or pheromones or different odor linked to either to conspecific or to particular pollutant, for example. They are also very visual animal, well, not all species, of course, but many of them are using vision. Of course, all the fish have locomotory ability to escape predation. So this is also something that we can measure. And yeah, the important point is that all these behavioral traits, we can actually imagine to measure them. So I will illustrate this later. Schooling behavior is also something very important. Reproduction, we have some fish that are very, some species are building nests, they are having courtship, so there is really a lot of traits that can be analyzed there. Spawning site selection and homing are also very important and can be modified by aquatic pollutant. And of course, all the social behavior can be detailed, like competition, aggression, etc. At the same time, they have also listed physiological mechanisms that we are known to underlie behavioral alteration. So, because of course, like I said, the first step is to detect, so to have a sensory perception. But actually, some pollutants, like for example, copper or cadmium, they will actually destroy the smell of the sense of smell, for example, because they are damaging the ciliated cells. So it's a direct action on the perception. Endocrine disruption is another one, like some compound will mimic natural endocrine or hormones, and then of course you will have some alteration, like sex change in fish is very well known and documented. You can have also neurological dysfunction, so you can have some compounds that act really directly on the nervous system. And also metabolic disruption, because like I said, some toxicants are metabolized, but of course this is costing extra energy to the body of the animal, so it's actually a costly activity. And this can create imbalance in how the animal is managing energy. So the whole idea here and the challenge actually is to be able to combine some aspects that are listed in the behavioral effect and some in the physiological mechanism. And you can imagine all the interaction that exists between these two small lists. So it's really a lot of things that we can explore and try to understand better. And then comes zebrafish. So I guess some of you are familiar with zebrafish or want to work with zebrafish or already have a long history of working with zebrafish. The beauty of this fish is that it has this development that is really in a transparent egg, so everything is really beautiful. You can observe the emergence of the creation of the animal, and because it's completely transparent and it's happening in a few hours, you can actually see, like here we see the construction of the eye, the construction of the muscle. So if we are speaking about using pollutants, either in directly, in exposure in eggs or in the mother, then we can imagine that something will happen, and we can actually see it at this stage, at this very early stage. Then, if we try to summarize everything that we can imagine doing, so here you have a family of different families of toxicants like metallic, organic, microplastic, pharmaceuticals, personal care products, etc., because there are always new ones. And in our zebrafish, of course, zebrafish is a small fish, so there are things that we can't do, but there are a lot of things we can access, like working, of course, on the behavior, but also metabolism can be studied, and reproduction, of course, malformation and growth, if you remind this transparent egg. And we can work on immunity, genotoxicity, tumor regenesis. So it's really a lot of things that we can explore. And all these traits or these phenotypes, we could say, you can interpret them either at the individual level, like it will act on individual survival, and you can also interpret it in terms of population maintenance, because of course it is linked. But if your individual is growing less, producing less eggs, for example, of course it will have a direct consequence on your population. So all this we can actually work on. And yeah, this was a small extraction of the number of papers that have been published over 20 years. It was really rapidly expanding. I think now it's more or less stable for the past 5 years, but I have to update this graph. But it has been really very rapidly expanding, both in ecotox and in toxicology. A bit less quick. And I did this word analysis of what was actually published, and it's actually quite interesting, because the word that comes the most often are the larger in size in this cloud, and you see that disruption, gene, and embryo have been the mostly used trade or material to say. So ecotoxicology was clearly developed using fish embryo for fish embryo test. Development is also something that has been very much used. And among the emerging, we have some focus that is more and more placed on behavior and hormones, but you see it's not that important. And the things that are really striking in what is really striking in this cloud, it's that clearly there are terms that are missing, like among all this paper, brain was not mentioned, neurotoxicity was barely mentioned. So it really means that we still have to do some progress, because as I just told you, that perception is the first thing. Detection of a threat or of toxicant is, of course, linked to perception and to central processing. And central processing is the brain. And neurotoxicity directly affects this perception and this central processing. So now if we go to the behavioral toolbox, so I will try to focus more now on behavior and how can we use it. So this is a drawing. You don't have to understand all the arrows. It's just to show you how complex it can be. If you want to study the nesting activity of a bird, you have to look at many behavioral units. And the beauty of it is that everything is actually measurable, and it can help you to understand how the animal is actually constructing something. So the tactics that the animal is using here, the bird, he has to find a place, he has to find the material to build a nest, then he has to find a mate. So it's step by step that this is constructed. And then the general strategy, the aim, is actually to find a mate and to make offspring. So, and all this we can actually do with our fish in response to aquatic pollutants. So, for example, we can work on larvae. So there are different tools that exist to actually study the behavior of larvae. So here you can study the behavior in plates. You can use petri dishes. You can use very simple system where you use a camera into a closed box, or you can use an expensive out-of-the-shelf made system. And using this, well, we will look afterwards what are the readouts. Or you can also work with juveniles or adult fish. You can work on different activities, on shoaling behavior, on maze exploration, preference place, and all kind of activities. So this is really a really nice toolbox that we have now. So, okay, we have a toolbox, but now we have question we want to tackle. So we have to know better which test or which protocols can be used for what, because this is what is important when you define an experiment, is to know your scientific question and then to know how you will reach your results. Like in this plate, you can actually monitor simply the swimming activity in your well plate. So you will have activity of your animals, and you can also use it to evaluate anxiety. And because you can actually measure the time the animal is spending in the center of the well or in the outer edge. You can also challenge the animal with a dark phase. So you suddenly shut down the light, and then you will trigger what is called a larval photomotor response. And this is also very much used to evaluate anxiety in your animals, how it reacts with hypo or hyperactivity. You can do very simple environmental choice with group of larvae. Just, you have a dark and a clear side, or you can use different colors. And this will help you to eventually distinguish scotophobic and exploration behavior in your animal in response to a pollutant, for example. New environment is also something you can do in 1 month old juveniles. So these are also still small zebrafish. So you can monitor the swimming behavior in petri dish over a certain period of time in the outer edge or inner edge of your petri. Same you can do a preference place, exactly the same as for larvae. You can do for 1 or 2 months old juveniles. You can adapt your setup. And then if you go to more like juveniles towards adults, you can actually do the same, new environment. And there is one test that is very well known, is novel diving test, the diving tank. So you will have, you put your fish in this setup and you monitor the time the fish is taking to actually leave the bottom to go towards the surface and occupy all the space. So this is also something that has been used to monitor activity and anxiety a lot. Maze, you can imagine all kind of shape of maze, and this one, for example, is really nice because it gives you an index of exploration. You just simply count the number of zones, and then you can evaluate exploration and risk taking, because the animal doesn't know where he is going and what is happening at the next corner. So these are really very simple methods for which you don't really need sophisticated equipment. You can actually analyze the videos without sophisticated software. And at the same time, because zebrafish is very much described in terms of genes and brain atlas, and all the endocrinology is known, you can, of course, sample your animal, and if you have access to facilities, you can monitor, for example, cortisol concentration, the stress hormone of fish, and try to evaluate if your animal is doing okay or is very much stressed. Do, of course, an endocrine profile like sex steroids, look at some genes expression, do in situ hybridization if you have all the information and the equipment. And you can also look at neurotransmitters, so go deeper into the brain and how the signal are transmitted in the brain. So this was all the concepts that I think are very important to master when you want to tackle this question of behavioral ecotoxicology in fish. You need to have all this information. And now I would like to illustrate everything I've presented before with some examples to show you concrete things. So, like I said, we work mostly on organic contaminants so far. So we have been using PAH. So PAH can be are very much widespread because they are, of course, due to mostly to human activity, because it's all the smoke from the fire or from the use of fuel or from all the human activity. So we have been using some compound extracted from light or heavy fuel and exposing animal through their whole life. Because what is also important in the context of ecotoxicology is to be as realistic as possible. So it's important to use concentrations that are actually found in the environment, to use exposure routes that exist. You don't make something that is completely out of reality. And fish can be exposed through their whole life. It's not sometimes it can be only for a period of time, but unfortunately, very often it's for the whole life. So what we have been doing is contaminating the animal from mouth opening to reproduction with sequential feed contaminated, and then we have been challenging our animals. Look, like here, for example, I have this example of the Z maize. So what we had is we had different compounds. So the green okay. So the fish was in the shelter before, and then we open the door, and then the fish can explore this Z maize. So as you can see, a zebrafish, even if it's a rather tiny one, is actually exploring back and forth and going all the way almost to the end of this maze. And on the right-hand side, you see the difference we had between a control population in green, where the animal was all of them, or most of 80% of them, were actually reaching the end of the maze. And whereas the fish that were contaminated with heavy oil, they were actually not exploring the maze and not moving so much. So we really had a weaker exploration for the fish that were exposed to heavy oil. And this has, as you can imagine, a direct consequence, because if a fish is not exploring its environment, it will be really impeded to find food or to find mate. So it will really impact on the resources it can acquire in the environment. We saw also, using the novel tank diving tests, that the fish can also have a very different response. Like here, so you put your fish gently in the tank, and this is a test that you do over 5 to 6 minutes, so it's a reasonably high throughput. And what is important to know here is that the normal reaction of a fish at the beginning in a novel environment is to stay close to the bottom. This is a very ancestral anti-predatory response. I don't know what is happening in this environment, so I stay close to a shelter, the bottom, and then the fish will progressively go all the way up. I accelerate here to the surface, where it's more risky because there can be a predator. So if we compare all the situation we had with the different PAH concentration and compound, we clearly had very big differences, and we had a lower activity and also more anxious fish than the one that were contaminated. And then we try to look at monoamines. So monoamines are a neurotransmitter that are really acting on the level of activity of a fish. And we looked at the concentration of different compounds, such as serotonin and dopamine and DOPAC, the metabolite of dopamine. And we clearly had differences emerging between our controls and our two light and heavy fuel compound. So globally, we had a reduction in the brain monoamine, and we wanted to know how is it linked to the behavioral response we see. So we actually investigated further, also in relation to the response to the diving test. We clearly had differences between our group of fish, and the increase in the fish that were more anxious also had less serotonin and neuroadrenaline. So it's really, we could really see a mechanistic explanation to our increase in anxiety, to our behavioral readout. So that was really interesting, because this is giving us possibility to go even deeper. And so we had this increase at the same time as the fish were more anxious. They were also showing an increase in diurnal activity, especially for the fish that were in this light oil treatment. And it was really interesting that we found that some people had published exactly something very similar in the mouse, and they had shown that dopamine was actually regulating ultradian rhythm. So the activity of the mouse can be regulated, and we actually, our hypothesis is that we have also the same, we observe the same in our fish. So some deregulation of the ultradian rhythm, and this is leading to hyperactivity in our animals. Okay, so now I will go to a second project to give you some other dimension going towards modeling. So this was a project where we wanted to look at the full life cycle of zebrafish and look at PCB and PBDE effects in particular. So they are also historical. PCB are historical contaminants, and PBDE are more emerging and still on the rise, where PCB are tend to decline in the aquatic environment. So for zebrafish, again, we expose the animal for all their life, so from egg to from larvae to reproducing adults. And our objectives were to evaluate behavioral and molecular responses related to anxiety in the exposed fish, but also in the next generation. And we looked at different consequences, and all of them, like endocrine disruption and behavioral disruption, reproductive disruption, all these have in common that they are very important for the maintenance of the population. Fishery people, they speak about recruitment. This is a contribution to the next generation. So here we used PCB, PBDE in a mixture. So we try to reconstruct a mixture similar to what is found in the Seine estuary, which is one of the most polluted European estuary. And we also contaminated the food, which is the main route of exposure of fish in the wild. And again, we added this to the food, and we followed the contamination throughout life from 5 days to 4 months. And of course, with zebrafish, you can also have a lot of fish at the same time. So we actually reared fish under this different control and mixture condition. And we had about 2,000 or 3,000 fish in different batches through 2 years. So we had a lot of data. We could gather a lot of growth and reproduction information. So here, as an example, you see we have been following some batch of fish over a year. So here you have the difference between this is a growth history of our fish. We looked at spawning probability. So we made pairs of fish in the evening, and we were monitoring whether the fish were actually spawning or not, and how many eggs, and if the eggs were fertilized or not. So here you have the cumulative number of eggs through time, and you clearly see differences. So the blue one is the control, and the contaminated fish, the F0, the parents, they really clearly produce less eggs through time. So this is really something that was really striking in this study. So it was a large project with all these aspects that were monitored. And to summarize, what we saw a lot of effects on life history traits. And like we had, in terms of survival, the mixture was actually not lethal at all, because it was environmental dose, of course. So we had no effect on survival of our F0. We had, of course, like we saw, a lesser growth, but surprisingly, and this is important for the next one of the next slide, the fish were actually growing more in length, and it is an important point. Body condition was not changed. Spawning probability was really decreased. Number of eggs was the same, but it was occurring less often. Fertilization rate was also the same. So here it's important to know that if you only count the number of eggs and the fertilization of the egg, you could say, well, that's not up, nothing is happening to the fish. But actually, it was acting on reproduction, because fish were not spawning as often as the control. So eggs were the same, but not the spawning probability. And the F1 larvae, they were dying more than the controls. And so we also had an effect on the F1. And Khaled, this PhD student, he was studying using all these data sets to build a model, because you can actually derive from this data on your zebrafish. You can develop all kind of models. And in this case, he used the depth theory. And to make it really short, the depth theory says that the food you are ingesting is assimilated and building up your reserve. And the reserves, they divide in two compartments: the structure, so the body growth, somatic maintenance, and the reproduction on the other side. And there is a sort of maturity maintenance to produce eggs, and also a reproduction effort, of course, when you release the egg. So all this can be parameterized and evaluated in terms of energetic cost. And what he found out, that the fish that were actually ingesting this mixture of PCB and PBDE, they were actually the difference we saw between the model for control and the model for exposed fish was actually this point number 3, the balance between giving energy to the soma or giving energy to the reproductive buffer. So, and this was really surprising, because this is very unusual that species are affected in this pathway. So the physiological mode of action, the PMOA, was actually this balance between growth and reproduction, and the share of energy was modified by the mixture exposure. And you can imagine all the consequences for the life history of the species. And here, in this case, again, we were also monitoring endocrine disruption, and we could actually relate the physiology, in this case, and the model information, because we actually saw that there was different concentration of beta estradiol, so sex steroid hormone or onschetosterone, between our fish from the solvent and from the mixture group. So this was also going right in the same direction towards an important endocrine disruption. And yeah, and in this case, this is the same information. But what also Khaled found was, what was also important is the cost of one egg. This E0 was actually increasing in our mixed fish, in our exposed fish. So there was an energetic cost that was increased to produce egg. And then what he did next, so that where I point out that the fish were growing more in length. What he did is, if we consider that all this data set could be the data set from an exploited population by a fishery, for example, what would happen to the animal? And if you recall, so we have, so he did a model where he simulated a fishing activity on our zebrafish population. And actually, the solvent fish, they were in a different. Here, if you look at the number, the lengths here. Oh, sorry. No, the abundance of the animal and the biomass, so the weight of the animal, was actually very different between the solvent and the mixed. And actually, the action of fishing, because when you fish, when you exploit a population, you tend to capture the larger fish. And actually, the mixture exposed fish, they were more sensitive to the fishing pressure. So we make the hypothesis that if this is happening really in an exploited population, then it's not only because the fish they have endocrine disruption acting on reproduction, and they are eventually caught in larger number by the fishery. So all the action on the fish are negative, and the MSI, this is the exploited population, is actually really more important for exposed fish. So over-exploitation risk is also increased. So they are more exploited and they reproduce less. So I'll let you imagine what is happening to the species. And very quickly, before I leave some time for questions, we also looked at transgenerational aspects because we were really curious to see what was happening to the offspring. And the beauty of zebrafish is that you can rear them for many generations over a short period of time. And in this case, we went from the parent, we had F1 fish that we reared without contamination, and then F2, F3, and F4. And we had a lot of interesting results, and I will directly go to the summary. So yes, only the parents were exposed, and then no more exposure. So we looked at different endpoints. We looked at larval response to the light and gene expression and novel tank in the adults. So we did anxiety evaluation. So we looked at different traits, and I will go directly to the conclusion, which is here. So in all these different generations, we saw modification of anxiety from in F1 and in F2, changes in locomotor activity, which was largely reduced in the F2 and after. So the fish exploration and locomotor activity were decreased. We also had changes in DNMTs and CFOs, which are neuronal markers. So brain activity was actually modified only by one generation exposure. So even if we had little effects in general in F0, except for growth and reproduction. In our ecosystem with all the human activities that is going on. Okay, well, I will skip through this because I've been too long. Oops. But just as a conclusion, yeah, I hope I have illustrated all the value of zebrafish and how it can really help us to tackle the many questions that are still there concerning ecotoxicology. And on top of this, if I didn't focus so much on it, but if you are working with the early life stages, the ELS, they also comply with the free air because nowadays they are not considered as experimental animals, so they are not considered in vivo but in vitro. Well, the regulation and the law will probably change in the future, but as for now, when we work with this early life stage, we can actually comply with the free air. And there are still much more things to do, like if you're when we are working, we can, we have to know the life history of our fish because it's also very essential. We could also look at coping style because this is also something that influences behavioral readouts. With zebrafish, just a recommendation, be careful about the fish strains you are using because wild types, transgenics, or different population of zebrafish, they are not all behaving the same. The degree of domestication and selection is essential to know. And keeping variability in the experiment is also sometimes a concern. So when you work with all the presentation I give you here on zebrafish, we always have an important focus on the spawns, to use many spawns, to mix our population and make sure we have something that represents life and what is happening in the environment. Okay, yes, this was the end of my presentation. I don't know how to go back to here. Yes.
Hello again.
Yes.
Thank you, Dr. Marie, for this marvelous presentation. Thank you very much for spending some time with us. We actually, until now, at least, we don't have any questions from the audience. I don't know if anyone wants to make a question really quick right now. But if not, I'm sure that if anyone think of a question later that can be asked directly to you, right?
Yeah, sure. Yeah, sure. No problem. That was, I think, at some point that was my my email. So don't hesitate if you
Yes.
need some advice. Okay. Well, thank you very much. Have a good day.
Thank you for staying with us. And everybody, please stay together because there's more lecturers coming today. So just wait for the next one, and we'll be right back.
My name is Mark Francis. I'm the founder of Aquaneering. For the last 20 years, Aquaneering has built a team of dedicated professionals to support your research. Our mission is simple: provide the highest performance zebrafish housing at a reasonable cost. Our team is here to allow you to focus on your research.
We at Alesco work daily to create solutions and equipment for the scientific community, contributing to the advancement of Latin America biomedical research. Our mission is greater than just providing equipment. It is to provide security with a quality service and mainly with enormous respect and attention. For this reason, we invest in high technology and seek to maintain long-lasting relationships. Our commitment is to be a reliable partner that understands the needs, the reality, and the conditions of each customer to always offer the best solution. We understand the benefits of scientific research for humanity, and this encourages us. If today we have a better quality of life, greater longevity, if we beat cancer or use a medicine for a headache, it is because the advance of biomedical research allows us to. We trust in the work of researchers, in science, and the scientific community, and we pride ourselves on doing our part. Just like you, we are passionate. Science is what moves us, because for Alesco, research is for life.
Welcome everyone to FST's presentation. We appreciate you taking the time today and look forward to helping you find the right tools for the right application and your endless discoveries and research. Find Science Tools has been in business for over 45 years, providing scientists, researchers, and other life science professionals with high-quality surgical instruments utilizing German steel and German craftsmanship with a secondary quality control inspection in our Heidelberg office. As we say, we only carry the best and offer a multitude of fine microsurgical instruments like spring scissors, forceps, and rongeurs, but also a variety of options for wound closures and animal identification. In addition to our 3 FST offices, we have over 50 distributors worldwide. While the majority of the audience is based in the USA, please note that we can ship anywhere in the world, and any products not listed in our physical catalog or online at findscience.com can be sourced as a special order, and we will work with you to get you what you need. Before we start, for those who are unfamiliar with FST, let me go over our core values we hold ourselves to and strive to provide to our customers. Quality. Impeccable product quality is what differentiates FST from the others. Secondly, our customer support. We strive for 100% customer satisfaction. Lastly, our QC department in Germany upholds our manufacturers to the standards FST and our customers expect with every instrument.
When you walk into a pharmacy, over 90% of the products on the shelf have been tested on animals, such as rats, rabbits, and primates. These animal models have the complexity one needs, but they are costly, time-consuming, and ethically challenging. Also, 9 out of 10 products tested in animals fail when administered to humans. Even with biotechnological advances, the use of these animals in product research and development is still massive. Is there no other alternative to the use of these animals? And to what extent is maintaining this model worth it? Today, regulatory bodies pressure companies to change this scenario. In this perspective, large companies such as Natura, Boticário, and others are adopting an alternative methodology to replace the use of animals in tests. Thus, to offer innovative solutions, ZSafe was born. We are committed to using fish chambers for the toxicological evaluation of new products. It is an innovative, alternative, and predictive model. The fantastic thing is that, despite not being considered an animal model, this embryo shows cellular activity, guaranteeing the same reliability in animals and greater than tests in cells. Our tests reduce research time and costs in new product development by more than 50%. We have the perfect formula to provide greater profits for companies and reduce any possible ethical issues regarding animal use and welfare. So, it's a win-win relationship. Our differential in the market is the competitive price, shorter service time, and delivery of reliable results. We have a multiple competent team that focuses on innovative solutions for the market and works to generate biomedical research safely and efficiently for the good of our customers, society, and animals. We are ZSafe, and we are swimming towards a safer future.
Situada no estado do Rio Grande do Norte, no Nordeste brasileiro, a Universidade Federal do Rio Grande do Norte está entre as maiores universidades da região. Fundada em 25 de junho de 1958, possui uma área construída de aproximadamente 300 mil metros quadrados, que compõe os 5 campi distribuídos por todo o estado. Tem hoje mais de 40 mil estudantes divididos em mais de 110 cursos de graduação, mais de 90 cursos de especialização, incluindo as residências, 80 cursos de mestrado e 40 de doutorado. Conta com mais de 2.300 professores, dos quais mais de 1.500 com doutorado. São mais de 1.900 projetos de pesquisas desenvolvidos pelos mais de 340 grupos de pesquisa cadastrados no CNPq, mais de 700 projetos de extensão e 4 programas permanentes, além de mais de 3.000 servidores técnicos administrativos. Como uma universidade de ampla atuação, as atividades de ensino, pesquisa e extensão se estendem a todas as áreas: ciências humanas, ciências sociais, ciências exatas e da terra, ciências biológicas, ciências da saúde, tecnologias e artes. Entre as áreas de destaque da UFRN podemos citar o Instituto do Cérebro, que realiza pesquisas na área de neuroengenharia para o tratamento de doenças vasculares, epilepsia e outros distúrbios neurais. O Instituto Internacional de Física, que promove a troca de conhecimento científico com a comunidade internacional em áreas estratégicas. O Núcleo de Petróleo e Energias Renováveis, que por meio de parceria com a Petrobras, é considerado um dos maiores polos de pesquisa na área, formando mais de 1.000 profissionais em níveis de graduação e pós-graduação. O Instituto de Medicina Tropical, que tem por objetivo o estudo de doenças infecciosas com maior incidência em continentes de clima tropical, como América do Sul, África e Ásia. O O Instituto Metrópole Digital, que atua na formação técnica e acadêmica por meio da inclusão social e digital de jovens, desde o ensino básico até a pós-graduação. A Escola de Música da UFRN, que tem por objetivo educar, produzir, disseminar conhecimento e fazer música como forma de contribuir para o desenvolvimento humano, promover a justiça social, a democracia e a cidadania. Nossa escola acolhe mais de 20 grupos e a Orquestra Sinfônica da UFRN, uma das únicas orquestras universitárias do país. O NUPLAN, Núcleo de Pesquisa de Medicamentos e Alimentos, um laboratório oficial destinado à produção de medicamentos para o Sistema Público de Saúde brasileiro. Entre suas atividades, o NUPLAN também oferece suporte acadêmico para a graduação e a pós-graduação, promovendo o ensino, a pesquisa, a extensão e a inovação tecnológica. O Ágora, Instituto de Línguas, Literaturas e Culturas Estrangeiras Modernas, que tem como principal objetivo oferecer cursos de língua estrangeira para estudantes, técnicos administrativos e professores da UFRN, bem como testes de proficiência para possibilitar a mobilidade internacional na universidade. O Ágora também recebe estudantes e pesquisadores das instituições parceiras da UFRN e de outros países, por meio do curso gratuito de português para estrangeiros. Na extensão, o programa de maior abrangência é o Trilhas Potiguares, que leva o conhecimento de professores e alunos aos municípios do interior do Rio Grande do Norte, em ações sociais desenvolvidas em parceria com as prefeituras municipais. Com mais de 20 anos de atuação, o sucesso desse programa já o levou também ao exterior, com a ida de nossas equipes a Moçambique, atuando com as equipes locais. Outra ação de relevo da instituição é a Semana de Ciência, Tecnologia e Cultura da Universidade Federal do Rio Grande do Norte, maior evento de atividades científicas, tecnológicas e culturais do Estado e um dos maiores palcos para a interface da UFRN com a sociedade. Em sua estrutura, a UFRN também dispõe de 3 hospitais universitários, 2 deles na capital e um no interior, 22 bibliotecas, 3 museus, canal de TV digital, rádio, agência de comunicação, editora, núcleo de arte e cultura, núcleo de educação infantil, residência e restaurante universitário. Em seu polo esportivo, disponibiliza campos de futebol e de treinamento oficiais, ginásios, piscinas olímpica e semiolímpicas. Assim, a UFRN busca a excelência e a inovação acadêmica por meio da formação de profissionais capacitados e ações educacionais comprometidas com a produção de conhecimento articulado com a integração social e comunitária, desenvolvendo soluções para os problemas regionais, nacionais e internacionais.
generate, and spread knowledge to form transforming agents. This is the goal of the biggest university of Rio Grande do Sul, State Northern Region, and one of the biggest from the country's southern region. With half a century of history, UPF maintains its roots in educational excellence, research and extension incentive, and the constant search for technological innovation. There are 9 campuses that are distributed around the region and 12 academic units, offering undergraduate, specialization, master's, doctorate, and postdoctorate courses. The infrastructure is a great differentiator. There are clinics, rooms for practical experimental teaching, laboratories, amphitheaters, auditoriums, and libraries with hundreds of thousands of titles available, both in physical and digital format. UPF stands out for the quality of teaching, academic, investigative in nature, the dialogue with the community, the performance in the job market of professionals who have graduated from this institution, and by the qualified teaching staff with teachers who promote the renewal of methodologies for the teaching and learning process. The research incentive in the university generates and disseminates knowledge. The extension reinforces the university's commitment to the community, with projects and programs that, through services provided, approximate academics to social reality and contribute to the sustainable development. Along with a university's social responsibility policy, the institution promotes citizenship, environmental awareness, social inclusion, culture, and development. Major events are held at the university. An example is the National Journey of Literature, one of the greatest literary manifestations in Latin America. The Radio UPF and UPF TV broadcasters with open signal and educational character provide the community with cultural programming focused on local development. Facilitating access to higher education, the university has several lines of credit, scholarships and funding, benefiting more than 70% of students. One of them is the PAI, UPF's own financing program. Every semester, possibilities of exchange are offered to students through agreements with institutions in several countries. Acknowledgements strengthen the greatness of UPF. The university is among the best private institutions in Brazil. Its courses received grades 4 and 5 in the assessments made by the Ministry of Education. The institution has also received in recent years other important awards, such as Social Responsibility, granted by the State Legislative Assembly. The Planalto Medio UPF Scientific Park contributes directly to the development of the region. Connecting innovation, science, and technology, the UPF Parque, with its technology-based incubator, supports and encourages innovative projects and local entrepreneurship. Providing quality of life, UPF offers to the academic community services that favor inclusion and permanence in higher education and the construction of great stories. The university has a community center, a leisure environment to eat, interact, and rest. This is the University of Passo Fundo, an institution that prioritizes ethical and humanistic formation. UPF, knowledge is our nature.
Hello, everyone. It's a great pleasure for me to present the next talk in this wonderful webinar. And it's my pleasure also to present Dr. Germán Zumbre, whom I must say I only met 2 weeks ago, and once Eve told me there was a free spot still in the webinar, I thought it was a great idea because I heard Germán's talk a couple of weeks ago, and it was really great. So I thank him for being here today with such a short notice. Germán is Director of Research Class 2 of INSERM. He's Group Leader of the Neural Circuits and Behavior Lab at the École Normale Supérieure from Paris, France. And this is from since 2008. He did his PhD at the Hebrew University of Jerusalem in Israel, and then went for a postdoc at the University of California in Berkeley, in USA. He has received several awards and recognitions, and also published several articles in the area he's going to present right now. So welcome, Germán. Thank you for being here. And I leave you to the presentation.
Okay. Thank you very much, Flavio and Eve, for the invitation. I'm very glad to be here. So in the lab, as all of you, we're using Zebrafish, and we're working on the dynamics of neural circuits and behavior. Just as a short introduction, I will explain why we're trying to study mainly the spontaneous activity in the brain. So several years ago, or just 3 decades ago, the work from Bonhoeffer and Miriam Greenwald using voltage-sensitive dyes in V1 in anesthetized cats show that V1 has a map of the different orientations that is organized in this sort of pinwheels, right? If you present an orientation as a visual stimulus in orientation like this, you see these white areas light up. If the orientation is different, other areas light up. But when you plot all the orientations together, you see this color. Every color is a different orientation. You see this map, which is always arranged about from this point, like all the different orientations are arranged there, turned in a sort of pinwheel around a given point, like this one that you can see here. So future work after several years, like 10 years after the first work, and the lab from Amir and Greenwald also using the same techniques. They showed that this is the evoke activity that I showed before, that the spontaneous activity in V1 resembles, as you can see here, that evoked by visual stimuli of different orientations. So this is interesting because until now, or until the 2000, it was thought that the spontaneous activity in the brain, especially in sensory brain areas, which was highly active, like if you remove all the sensory stimuli around an animal and you record from a sensory area, you will see that it's highly active. And people thought that this activity, ongoing spontaneous activity, is actually just biophysical noise that emerges from the circuit. And the brain actually needs to get rid of this spontaneous activity. But this was the first work showing that the spontaneous activity has a special temporal pattern, which suggests that it's not noise, but it's something else. And since at this time, the group of Amir and Greenwald was working with anesthetized cats and voltage-sensitive dyes that don't have cellular resolution, it was difficult to understand what was the biological meaning of this spontaneous activity and what are the mechanisms that allow the emergence of these special temporal patterns. So these are exactly the questions that we were trying to answer using the zebrafish larva. what's the biological relevance of the brain's ongoing spontaneous activity, and what are the mechanisms that allow the emergence of these patterns from the intrinsic dynamics of the circuit. So this I don't have to introduce, but just for us, for neurobiologists, the zebrafish is a very useful model, since allows us to record from the whole brain with single cell resolution in a behaving intact vertebrate. And also their behaviors are complex but still discrete, but allows it's very easy to analyze them compared to more complex mammals. We are using transgenic fish, like this one, that is expressed in all neurons using the LAV3 or HUC promoter. The CHICAN, for those that they don't know what's a CHICAN protein, it's a GFP bound to a calmodulin, so allows that every time that there's an action potential in a neuron, the calmodulin will interact with the calcium, and that will change the conformation of the GFP. So activity of the neuron will change the fluorescence of the GFP. The more fluorescence, the more active is the neuron, and we have a readout of the neural activity by looking at the fluorescence of each of the neurons. So for this work that I'm going to talk today, we use two-photon microscopy. We presented visual stimuli and recorded spontaneous activity while monitoring the brain activity. The larva is in a drop of agarose, so we can see the movements of the tail, and the animal is completely intact and anesthetized, so it's behaving almost normally, let's say. We focus mainly in this area called the optic tectum. The optic tectum is the main visual center in zebrafish and all teleost. It's the homologous to the superior colliculus in mammals, and its main function is to detect visual stimuli, mainly the spatial property, the spatial position of the stimuli, their size and orientation of the movement, and generate goal-directed behaviors, for example, to catch prey or to escape from predators. So the first thing we did is to look how this area, the optic tectum, responds to visual stimuli of different positions. So what we did is to present small light dots of light in front of the larva at different positions, like from the lateral right to the lateral left, passing from all the frontal areas. In this figure, I hope you can see my cursor. This is the midline, this is the cerebellum, the eyes are around here, and the tail somewhere over there. So when we present a stimulus on the right lateral part, the neurons in yellow were those that were responsive. If we move a small dot of light towards more central frontal areas, like -50 degrees, you can see that these active neurons, they move forwards toward more rostral areas. When the stimulus was right in front of the larva, you see these regions now involving both hemispheres. And if you pass towards the other, with the stimulus to the other hemifield of the visual field, you can see that now the other hemisphere of the optic tectum responds, and the center of gravity of the responding cells moves more caudal. And indeed, when you look at the central of gravity of each of these visual stimuli, the color now represents the position of the stimulus, and the dot represents the center of gravity of the visual responses in the optic tectum. You can see these graded change in colors, suggesting that in the optic tectum exists a retinotopic map arranged along these two axes. Okay, these areas respond to the frontal visual field, and these areas respond to the lateral field of view. When we look at the responses of the tactile neurons to the different directions of the stimuli, we observed here, for example, yellow is direction selective neuron that will respond towards down-up movement, green to the right, left to the red to the left, and blue downwards. You can see that there's no map of the direction selectivity. The direction selective neurons are completely intermingled within the tactile circuit. Then we looked at the spontaneous activity of this of the network of the optic tectum. Well, here, for those that they're not familiar with raster plots, here every row represents one neuron a long time. Here is one hour recordings, and the dots represent significant calcium events, moments in which the given neuron was active significantly. And at the bottom, you see the sum of all those events at a given time window for each frame in this case. So you can see that there indeed periods where there's synchronous activity within the network. Okay, some that are highly active, many neurons active at the same time, others, but it's a pattern that remains all along this hour of the recording. But it's difficult to understand what's going on and what's the meaning, biological meaning of these synchronous events. Even if you zoom in, you clearly see now these synchronous events across many neurons, but it still doesn't give you any more information than that. So for that, we use a sort of clustering method, a method based on PCA and factor analysis. We can discuss more if you are interested afterwards. I won't go into details during the talk, but this method allows to group neurons that they tend to have a same activity history of their activity, that they're active when two neurons are active, these covariate and will be grouped within the same group, the same cluster. And this is when the same raster I showed you before, when we order it according to these different clusters they belong to, you can see that below the red line, there are neurons that are completely isolated from the rest. They don't belong to any group, but you can see group of neurons like this blue group that at different time points they tend to be active together. Or the red one here and 4 times within the hour, or this blue these 2 times. And you can see that our method actually allows to see overlap between the groups. And indeed, there are certain neurons that belong to more than one group. For example, these neurons here, at one point they belong to the black ones, and at a different time point they belong to the blue ones. Then I don't know if it's possible to interrupt me, but if there's something not clear, maybe Flavio can stop me and clarify that. Since we were using calcium imaging, we could go back to these assemblies and ask where the neurons of each assembly are placed within the network. What's the topography of these assemblies? And you can see that most of them, they were quite compact. There were many neurons that were one close to the other, to the neighboring ones. And we call them retinotopic light because they resemble those induced by visual stimuli of small size. But there were others that were more sparse, sometimes involving both hemispheres or just one. And here you can see a movie of, this light sheet microscopy, where you can see in the optic tectum the emergence of these assemblies. In red, you see neurons that are more active. The activity of the neurons of the changes in fluorescence are labeled in red. So you can clearly see by eye even the emergence of the assemblies. This movie is faster than the real time. It's like 10 10 times faster. So these assemblies emerge slower, but more or less with the same patterns that I just showed you. So when we color the neurons in the optic tectum that respond, so we color them according to the stimulus they respond to, you see this graded change in colors showing this retinotopic map that I showed you before. So these neurons respond to the lateral sides on the left side, and these violet ones just in front of the fish. And when you do the same thing with in the same optic tectum of the same fish, and you color the neurons now according to the assemblies, to the groups of neurons that fire together, they're active together. Here the red ones, for example, they were active at the same time during the spontaneous activity periods. You see the very similar graded change in colors, suggesting that this spontaneous activity on the tectum reflects or recreates the retinotopic map. Okay. Then to understand what's the biological meaning of these assemblies, we ask which type of neurons, functionally type of neurons, are grouped in these assemblies. And for that, we look at the correlations of the spontaneous activity. So higher correlated neurons will be those that belong to the same assembly, compared to the similarity between the tuning curves. How well two neurons respond to the same position of a stimulus in the field of view. For example, two neurons will be highly correlated in this axis if they respond exactly to the same position of a light dot in the field of view. And what we saw, there was a bias for higher correlation values. So what does it mean? That neurons that are highly correlated spontaneously, also they share the same functional properties, okay? The black line is just a new model to see what more or less is significant or not. And this bias was indeed significant. In contrast, when we did the same analysis, but now rather than looking at where in the field of view whether the visual responses are similar, we look at the direction selectivity of the neurons, and we saw that there's no bias for the direction selectivity, meaning that the assemblies do not group these cells that share the same spatial tuning curves, but not necessarily the direction selectivity of the neurons. Meaning that within this group of neurons, you have neurons that respond to the same place in the field of view, but not the same direction of the movement of the stimulus, the visual stimulus. Then we looked at here we plot the tuning curve of each of the assemblies. For every row is one assembly, and the tuning curve of the assembly is the sum of the tuning curves of each of the neurons within the assembly. So it's just an idea to see where that assembly responds to in the visual field. And you can see a diagonal here. Here is the azimuth, the position in the field of view. Every row is one assembly, and the color is the amplitude of the tuning curve. You can see that there's a diagonal, and that means that across all assemblies, all positions in the field of view are represented, okay? But if you do the average across all these assemblies, you see this in this color curve, you see that the lateral regions, okay, minus 50 and 50 degrees in the field of view, are more better tuned than those in the frontal parts. And also here it's this color, that is the color of the line. You see the red is also on the sides and more blue in the middle, suggesting that the activation level is how often these assemblies emerge from the spontaneous activity. So the assemblies on the lateral sides are better tuned and emerge more often. So there's an overrepresentation of lateral sites in the optic tectum compared to those in the frontal part of it. And to understand what's the biological or behavioral meaning or relevance of these assemblies, so we'll look at the prey catch-up behavior of the larva, which is mainly controlled by the optic tectum. Here is the larva, here is the prey, one paramecia that most probably all of you know. I don't know if it, well, it's not working. But anyway, in the movie, what you see is that this larva detects at one moment this paramecia is slowly moving and will generate prey capture behavior towards the paramecia. And when we calculated, what was the angle of the larva of the zebrafish compared to the position of the paramecia, and we found that most of the cases in which the larva first detects the paramecia, this was on the lateral sides, either left or right, and not in the frontal areas. And this is represented here in this polar histogram, where in red you see the behavior, the detection of the paramecia. It's better detect on the lateral sides. And in gray is what I showed you before from this curve, that the so the neural assemblies are overrepresented on the lateral sides. So this representation of the assemblies, overrepresentation of the assemblies, is correlated quite well with the behavioral prey capture behavior, the detection of prey during prey capture behavior. Then we'll look what we asked whether these assemblies represent stimuli of different sizes or one specific size. And for that, we test them with visual stimuli of different sizes, 4, 8, or 12 degrees. And you can see that all sizes are represented in this histogram, but those of 4 degrees are overrepresented. And then we ask whether there's a correlation with the behavior again. And we measure the distance between the larva and the paramecia at the moment of detection. And what we found is this histogram that shows that there's a peak at 5 degrees. Very similar what we found in terms of the assemblies. So what we suggest is that the connectivity of the tectum that creates these assemblies actually is tuned to better recognize stimuli, that vital stimuli, for example, prey of the larva. To understand the mechanisms by which these assemblies emerge, we did the following analysis. We calculate the activation probability of the neurons within the assembly. This is the topography of one of the assemblies in the optic tectum. This is another, etc. And we correlated with the correlation coefficient. This correlation coefficient shows how well at each given moment the network, the topography of the network looks or mimics the one of the assembly. So when the correlation is high, it means that all the neurons in the assembly are active and none outside the assembly. And when it's low, most of the neurons outside the assembly are active and none within the assembly. And what you can see is this exponential or explosive correlations in all cases, suggesting that as soon as a few neurons within the assembly are active, that will recruit all the others in a sort of preferred network state or other non-activations. Either the assemblies are active or non-active. Then we'll look at the interactions between the assemblies. Here, we presented, in order to force them to interact between each other, we present a small light dot. We saw the responses, then we presented another slide dot, small light dot. We record the responses, and then we compare these two responses when we present both stimuli at the same time. And we ask whether the response to both stimuli is the sum of both or something else. And this is what we got. This is a raster plot. Now every row represents one trial of this stimuli. In x-axis, you see the neurons we recorded from. So you can see if we presented a rostral stimulus, this group of neurons were active. If we presented the other visual stimulus in a different position, this group of neurons were active. And when we presented both here in yellow, rather than seeing the sum of both, neurons here and neurons here, we saw this red group of neurons that were active, suggesting that the interactions between assemblies is sort of winner takes all, suggesting that there's inhibition between them, and only if both assemblies are active at the same time, only one will win, will be active, and that will suppress through inhibition, the activation of the second one, of the nearby one. Then, to understand what's the behavioral function of these assemblies, one needs to actually disrupt the functional connectivity, break apart these assemblies, and look how the visual responses and behavior happen in that case. But this is impossible because if you disrupt the intrinsic connectivity of the tectum to synaptic blocking of the synaptic connectivity, then you won't be able to test the system by presenting visual stimuli. There will be no responses, of course. It's impossible to disconnect the presynaptic inputs, disconnect the postsynaptic inputs without disconnecting at the same time the presynaptic ones. But to our surprise, we found a mutant that was created in the lab by a postdoc several years ago, and we never used it. But at one point, we tested the spontaneous activity in the optic tectum. This is the wild-type fish, something that I show you, a raster plot that I showed you before, where you see these spontaneous activations of the assemblies. And when we do the same experiment in the MECP2 fish, you see it's a little bit more gray, suggesting that there's more spontaneous activity, and you can see it here in the baseline of the red curve here than compared to this one, but there's no synchronous events at all. So when we study a little bit more these properties of the spontaneous activity in both cases, you see that the frequency of events is higher in this mutant, in the MECP2. By the way, the MECP2, it's a human mutation that is found in Rett syndrome patients. We can discuss more afterwards, but I will focus here on the spontaneous dynamics mainly. So the duration of the calcium transients is higher in the wild type, shorter in the MECP2. But for us, the most interesting thing was when we look at the pairwise correlations, how well neurons are active together. You can see it here in this matrix of correlations where you see each row represents one neuron, and in color in yellow shows neurons that are highly correlated. For example, this neuron here is highly correlated with this neuron here. Okay. And blue means neurons that are not correlated at all. And if you compare the wild type with the MECP2, you see that MECP2 is almost all blue, suggesting that there's no correlations between neurons. There's no functional connectivity of the optic tectum. It's like all cells are completely isolated. You can see it here, the significant correlations between neurons in the wild type and in the MECP2. So for us, We didn't look what's the consequences of Rett syndrome, or we didn't try to study Rett syndrome through these mutants, but for us, this mutant was the ideal control to study the biological relevance of these tactile assemblies, neuronal assemblies that I showed you before. So then we presented visual stimuli to the wild type and to the MECP2 at different light intensities. So very weak, 0.1 represents a very weak light, and 1 represents a very strong light with high contrast. And in each graph, it's a raster plot of the visual responses. Each row is one neuron. The red thin line represents the onset of the visual stimulus. And just from seeing the size of these raster plots, you can see that there are more neurons that respond in the MECP2 to the visual stimulus than in the wild type. When you look at the average across all these rasters, you can see that when the light is very weak, neither the wild type nor the MECP2 neurons respond. And they begin to respond at around 0.3 of contrast. And you see that the response of the wild type in blue is relatively larger, despite there are less neurons responding to the stimulus, and remains larger almost throughout the entire experiment, the entire different intensities of light. Here you see the summary of these responses in terms of the amplitude of the calcium transients with respect to the light intensity. You see that for all light intensities, the wild type also has larger amplitudes of response than the MECP2. And the probability of response is also higher. How many times the given neuron respond to the stimuli represent stimuli of many times. So if it's responds to all times, it will be 1. And if it responds to half of the stimuli presented, will be 0.5. So the probability of the response is always higher in the wild type compared to the MECP2. So the response is more reliable. But in contrast, when we look at the number of cells of the optic tectum that respond significantly to the visual stimuli, we see that for the MECP2 was always higher than the wild type. And this suggests that the neural assemblies enable a spatially restricted visual response, so there are less cells responding, which is more reliable and with lower response thresholds. So it's able to respond in the wild type to weaker stimuli than in the MECP2. Then we ask whether the optic tectum from the the responses in the optic tectum, we can decode the presented visual stimuli. What does it mean? That if we can, from the readout of the activity in the optic tectum, we can actually understand which type of stimuli was presented. And we did that using a maximum likelihood decoder and represented here the results in a confusion matrix. But you have to know that here is the presented stimuli, and here is how well was classified by the maximum likelihood decoder. What does it mean? That if the diagonal is black, the decoder classifies perfectly well the responses according to the stimuli that was presented. If the areas outside the diagonal are white, it's good. If they're gray or black, it means that the decoder is not working well. And what you can appreciate here, the diagonal is much more clear in the wild type than in the MECP2. And you can indeed better decode across all fish the visual responses, the position or the intensity of the light from the recording responses in the optic tectum better than in the MECP2 one. We did the same thing with the position of the stimulus. Rather than checking or studying the intensity of the light, we presented stimuli at different positions, and then we asked whether from the activity in the optic tectum we can recreate the position of the stimulus that was presented. In the wild type, here are the rasters of the average response to the stimulus when the stimulus was 15 degrees, 35 degrees, 40 or 50 different positions in the field of view. And you can see that the responses, the neurons that respond are quite different. But when you look at these rasters in the MECP2, they all, except for 15 degrees, they all look quite the same. And in the confusion matrices, this is what you see. The diagonal is more clear in a wild type than in the MECP2. Again, from the responses in the optic tectum, we can significantly state what was the position of the stimulus that was presented that induced that response. Okay. And to conclude, so we went to the behavior. We actually we filmed the larvae when they were eating, and we calculate how many paramecia were eaten a long time. And what we saw is that at the very end, both fish eat exactly, the mutant and the wild type eat exactly the same. So at the end, both are have eaten similarly. But at the very beginning, when they're probably more hungrier, the wild type is able to eat more than the MECP2. And what happens is because it misses the paramecia, but because it doesn't see well or something else. So when we looked at the abortion rate, it means that the larva is trying to find paramecia or it was already engaged in the prey capture behavior, and at one point gives up because it is not able to find it. It wasn't the case, was not a significant difference between both. But when we look at how many bouts, how many correction movements they need in order to catch the prey, we found that indeed the MECP2 needs more corrections of the prey capture behavior before it gets the paramecia. So suggesting that probably the larva is not seeing well and therefore need more correction maneuvers. Okay, so just to conclude, today I show you that the ongoing spontaneous activity in the optic tectum has a relevant structure for the visual system. The structure mimics the visual induced responses and it's organized according to the tectum's functional retinotopic map. The spontaneous activity structure reflects the tectal connectivity based on a preferred local circuit with attractor-like dynamics. And this mechanism may improve the signal-to-noise ratio for better detection of specific stimuli of 4 degrees, 5 degrees, like the paramecia in this case, in low contrast or cluttering backgrounds. And that suggesting that the tectum may work as a virtual phobia that will improve the spatial resolution and improve the decoding of the stimuli. The tectal functional connectivity supports a restricted visual response, so there are less neurons that respond to the visual stimuli, but this response is larger and more sensitive, okay, and more reliable. What improves the decoding of the visual stimuli and enhances prey capture behavior. And so we suggest that these assemblies may act as a top-down phobia to increase the signal-to-noise ratio and improve visual spatial resolution, specifically for vital visual stimuli. Okay, and with that, I want to thank all the members of the lab, especially Thomas Pietri, who worked on this, Martin, who worked with the MECP2, and Sebastian Romano, who's not in the picture. He left. He's back in Argentina, who made the initial work. Okay, thank you very much for listening.
Thank you, Germán. We are waiting to see if there are any questions in the chat. I think there are no questions so far. So I can ask you maybe. So what I understand MECP2 is essentially a chromatin modifier, right? So it's a protein related to chromatin.
No, it's a regulator of expression.
Right.
So it's something very, very general. Yeah.
Yeah, exactly. It's very general. So what do you have any hints or have any idea, or are you trying to explore what could be doing at the molecular cellular level? So what are the targets here?
Yeah. So the thing that we are trying to do now is to do RNA-seq of the optic-tectum specifically, and compare the patterns in where there is some sort of regulation in the expression of certain genes that may be related to synaptic connectivity in the wild type and in the mutant. But it's going. We are doing that right now. We have no result.
I think they are not. Sorry about that. That's my dog. Bad timing for singing. Okay, so I think we have no more questions. Just I will ask people if they see any other question that I cannot see, but I don't see any in the chat now. Apparently not. And do you think that this? So you mentioned something like a top-down phobia activity for these assemblies and for this way of working of the tactile activity. Do you think this is only in the visual processing aspect, or maybe there is also that somehow could be also affecting how neurons in the retina are organized or connecting or eventually sending the stimuli to the tectum? So it could be really an effect on.
As far as we know, there's no back connectivity to the retina from the tectum. Although we have some clues from experiments that we couldn't explain, where our results could be explained only by that. So we don't know yet. We know that other modalities, sensory modalities, yes, they project, the olfactory system projects to the retina. So it's a possibility. But here, what we're trying to understand is not that there's an effect directly to the retina, but there's a sort of limitation of the visual response in the optic tectum. So restricting the visual response in the optic tectum, what it will increase. There's a lot of lateral inhibition between the assemblies, and what will reflect a sort of improvement of the spatial resolution.
Yes.
Yeah.
I got that. I was wondering if maybe even through, I don't know, trophic interactions, retina ganglion cells or something like that. That's I know an extremely open thing, an open field. Okay, I think there are no more questions. So I thank you, Germán, very much for your participation.
A pleasure.
And
okay, bye.
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 Thanks.
My name is Mark Francis. I'm the founder of Aquaneering. For the last 20 years, Aquaneering has built a team of dedicated professionals to support your research. Our mission is simple: provide the highest performance zebrafish housing at a reasonable cost. Our team is here to allow you to focus on your research.
We at Alesco work daily to create solutions and equipment for the scientific community, contributing to the advancement of Latin America biomedical research. Our mission is greater than just providing equipment. It is to provide security with a quality service and mainly with enormous respect and attention. For this reason, we invest in high technology and seek to maintain long-lasting relationships. Our commitment is to be a reliable partner that understands the needs, the reality, and the conditions of each customer to always offer the best solution. We understand the benefits of scientific research for humanity, and this encourages us. If today we have a better quality of life, greater longevity, if we beat cancer or use a medicine for a headache, it is because the advance of biomedical research allows us to. We trust in the work of researchers, in science, and the scientific community, and we pride ourselves on doing our part. Just like you, we are passionate. Science is what moves us, because for Alesco, research is for life.
Welcome everyone to FST's presentation. We appreciate you taking the time today and look forward to helping you find the right tools for the right application in your endless discoveries and research. Find Science Tools has been in business for over 45 years, providing scientists, researchers, and other life science professionals with high-quality surgical instruments utilizing German steel and German craftsmanship with a secondary quality control inspection in our Heidelberg office. As we say, we only carry the best and offer a multitude of fine microsurgical instruments like spring scissors, forceps and rongeurs, but also a variety of options for wound closures and animal identification. In addition to our 3 FST offices, we have over 50 distributors worldwide. While the majority of the audience is based in the USA, please note that we can ship anywhere in the world, and any products not listed in our physical catalog or online at findscience.com can be sourced as a special order, and we will work with you to get you what you need. Before we start, for those who are unfamiliar with FST, let me go over our core values we hold ourselves to and strive to provide to our customers. Quality. Impeccable product quality is what differentiates FST from the others. Secondly, our customer support. We strive for 100% customer satisfaction. Lastly, our QC department in Germany upholds our manufacturers to the standards FST and our customers expect with every instrument.
When you walk into a pharmacy, over 90% of the products on the shelf have been tested on animals, such as rats, rabbits, and primates. These animal models have the complexity one needs, but they are costly, time-consuming, and ethically challenging. Also, 9 out of 10 products tested in animals fail when administered to humans. Even with biotechnological advances, the use of these animals in product research and development is still massive. Is there no other alternative to the use of these animals? And to what extent is maintaining this model worth it? Today, regulatory bodies pressures companies to change this scenario. In this perspective, large companies such as Natura, Boticário, and others are adopting an alternative methodology to replace the use of animals in tests. Thus, to offer innovative solutions, ZSafe was born. We are committed to using fish chambers for the toxicological evaluation of new products. It is an innovative, alternative, and predictive model. The fantastic thing is that, despite not being considered an animal model, this embryo shows cellular activity, guaranteeing the same reliability in animals and greater than tests in cells. Our tests reduce research time and costs in new product development by more than 50%. We have the perfect formula to provide greater profits for companies and reduce any possible ethical issues regarding animal use and welfare. So it's a win-win relationship. Our differential in the market is the competitive price, shorter service time, and delivery of reliable results. We have a multiple competent team that focuses on innovative solutions for the market and works to generate biomedical research safely and efficiently for the good of our customers, society, and animals. We are disease safe, and we are swimming towards a safer future.
Good afternoon, everyone. My name is Claudia Carraro. I'm a PhD candidate from the Department of Biochemistry here in the Ribeirão Preto Medical School at the University of São Paulo. I would like to invite you to welcome our next presenter, Professor Sofia Oliveira. She's an assistant professor from the Department of Developmental and Molecular Biology and the Department of Medicine in Hepatology at the Albert Einstein College of Medicine in New York, USA. Her lab studies neutrophils and their role in disease progression, and they are primarily interested in unveiling how the presence of systematic inflammation and metabolic regulation found in middle inflammation and inflammaging settings alter these neutrophils. So feel free to send your questions on our comments here because they will be asked by the end of the presentation. Okay. And today's presentation is entitled A Glimpse of the Liver with Zebrafish. So welcome, Professor Sofia.
Hi everyone. Thank you, Claudia, for such a nice introduction, and thank you to the organization of the Zebrafish webinar to have me here again to be able to showcase what my lab is doing. We're a new lab at Albert Einstein, and this time I'm going to talk about the research that we've been developing in the liver disease field. So my talk for today is named like a glimpse of the liver with Zebrafish, and I really hope that you can enjoy all the things that we can do with this amazing model to study liver diseases. So Zebrafish has been an established model to study liver disease. We know that it has a similar hepatic cellular composition, functional signaling, and response to liver injury. We know that the cellular processes that mediate liver disease are conserved with mammal models, and it also shows similar genetic signatures in the several liver diseases that this model has been used in the last almost 3 decades. So as you can see in here that I'm showing you, we can easily use transgenic lines that we can use to image hepatocytes, biliary cells, endothelial cells, hepatic stellate cells, and even neutrophils, macrophages, and T cells, all the immune cells that we are used to see in the liver context. So, however, for many years, the liver, how similar it is, the liver architecture about, between zebrafish and mammals, and that has been challenging our field and the progression of, and to translate our findings in the liver disease field to mammalian systems. So I think this study has recently, it was a 3-dimensional analysis of the vascular configuration in the liver, and I think it brought some light into some of the questions that we had about the architecture of the liver. So we actually know nowadays when we compared mouse and zebrafish livers, there are some things that are similar and other ones that are quite different. Like, for example, this study has shown that the zebrafish liver has lobules with polygonal structures in which the central veins and portal veins are located at the center and periphery respectively. It also has shown that sinusoids connect the portal veins and the central vein and the portal veins. However, it has like these conserved architecture. There are several other things that are different that need to be in consideration when we are, expanding and using this model for research, such as, for example, the location of the intrahepatic bile duct is quite different between mouse and zebrafish. Only periportal in the mouse, in the zebrafish is kind of like randomly. The number of independent portal vessels that enter the liver in mammal, in the mouse, is a single portal vessel. In zebrafish, there seems to be many. There are also the wild transport cells in the hepatic lobe. There are differences, and also the hepatic zonation may be also something to have in consideration, because up to now, there were no targets that we can see in the mouse that show the same kind of patterns in the zebrafish. So, but with all of these, the zebrafish is actually from what we do in our lab, which is we are very interested in the role of neutrophils in the pathophysiology of NAFLD and other liver diseases. So, because the liver is such a deep organ in mammals, it is really hard to image and to study and to convolute the cell-cell interactions that occur between immune cells and even with immune cells and hepatocytes or other cells that might be present in the liver microenvironment. So as you can see in the zebrafish, it's quite easy using transgenic lines that we can really take a deep look into the liver microenvironment, and we can actually study these interactions. Like, for example, in these time lapses taken from a movie, you can really appreciate how neutrophils in yellow and macrophages here in blue are actually in contact for many minutes, and actually sometimes even hours. And in this case, we are very interesting, for example, in exploring and the convolute how neutrophils can crosstalk with other immune cells. And this is an extremely and powerful tool for us, aligned with the manipulation that we can use in the zebrafish model, as we all know at this point. Another key aspect for our research is that we are performing our experiments in a whole animal context, which, besides allowing us to track all these amazing immune cells, in this case neutrophils, it also allows us to study the metabolic disease or liver disease in the context of a whole animal approach, which I do feel is extremely important if we consider that the liver crosstalks with many other organs. For example, we all are aware of the gut-brain-liver axis and how the gut and the brain are involved in the development of liver disease. So if we think about liver dysfunction and how that dysfunction starts to produce hepatokines and many other factors that are released to the blood flow and how they can impact other organs, we can really appreciate how it is important to have a whole animal approach to study liver diseases. So today I'm going to talk a little bit about three projects that we have been developing in the lab in the last year. So the first one I'm going to talk is a really new project that it was from a student that came from Victor Mulero's lab that you heard yesterday. Was my PhD mentor. So Irene came and she did an amazing job for these 6 months, and we really appreciate for that. So I'm going to talk a little bit about that. I'm also going to talk a little bit about a project that was already published on Journal of Pathology from my postdoc, where we study non-alcoholic fatty liver disease associated hepatocellular carcinoma. And later, I'm going to really show a little bit how, like, the only model that exists to study fibrolamellar carcinoma, ultra rare type of liver cancer. So we have been publishing already a few papers on the liver disease field, and one already from our lab. So if you want to know a little bit more about our tools, go to our Jove protocol. The video is about to come out, and also all these other papers that we have published already. So let's talk about a little bit how these xanthine dehydrogenase deficiency can alleviate the diet-induced non-alcoholic fatty liver disease. And in this case, we're looking more to non-alcoholic steatohepatitis. So, but what is NAFLD and NASH? So NAFLD is actually the most common chronic liver disease in the world. It is a medical challenge that is becoming the leading cause of liver transplantation. It's actually a histological spectrum of pathological conditions that can range from simple steatosis to non-alcoholic fatty liver disease, as you can see in the stage B here in this diagram. But it progresses with time, and this takes several years. Can progress to more aggressive inflammatory forms that are called NASH, or non-alcoholic steatohepatitis, that eventually a percentage of these patients progresses to fibrosis. Some of them can go to cirrhosis, but the more interesting thing about NAFLD is that a huge amount of the patients that are being detected at the clinic, they actually go to hepatocellular carcinoma without going to cirrhosis, which really makes this disease kind of concerning, because these patients are not being detected early on and they are being caught like at really late stages. So there is an overall prevalence of 32.4% in the world. As you can see in here, this is something that is spread all around the world. It's not specific to a few countries. We think it has the association of NAFLD with other comorbidities, such as type 2 diabetes and obesity, which are actually that have reached epidemic levels all around the world, are the number one cause. Why are we seeing this growing alarming, this growing rate that is in the incidence of NAFLD? It is an unmet global public health and an economical burden to many countries around the world. And it seems to be significantly higher in men than women. And it has several triggers that, for example, genetic, but also nutritional and behavior contributors. In our lab, we're particularly interested in diet, but also in the mutation PNPLA3 I148M, which has been associated to the development of NAFLD in the Hispanic community. And since the Bronx is such a good niche, Albert Einstein is localized in the Bronx, so we are a very good niche to even explore these further into humans. So as I was mentioning, NAFLD pathogenesis is a problem. It's a worldwide problem. It's actually a very complex disease to target. That's why it's an unmet medical challenge. We can see that it's a very complex pathogenesis that will probably, and we believe deeply that it's going to need a combination therapy approach. And for such, it is important for us to understand how metabolism, oxidative stress, fibrosis, immune response, how all these pathways are actually coordinated, to understand the pathophysiological mechanisms that are involved in FLD and its progression, so that we can not just target, but also target depending on the stage that we are at. So in our lab, we have been using a NASH diet-induced model, so a model that allows us to reach these steatohepatitis very quickly. We feed the fish with a high cholesterol diet for about a week. And we, if you want to know more, go to our Jove protocol. But we are also trying to develop at the same time right now in the lab. We have a collaboration with Sparos, with Annabella Van Simon Brito, John Rawls, and Stephen Farber. We are trying to develop reference diets, high fat, high cholesterol, but also Western diets with high fat, high cholesterol, and fructose. So diets that will help the community to come and use a similar tool to develop these metabolic diseases and to help us to reach out a consensus about the standards that we need to develop disease and push forward the field. So stay tuned, because there's more to come in the following year. And we already have these diets and we're already testing them in the lab. And this is extremely important for us for our future work, that we really want to be able to study progression of the disease. But the model I'm going to talk today is an acute NASH model. So it's just 1 week, we can really reach that inflammation and liver injury, and allows us to really study the microenvironment in a very short period of time. So as you can appreciate in here, just by taking the fish and putting them under a microscope, we can really see these accumulation of fat to start to occur in the liver. We don't need any kind of lipid probe to look at it. But of course, if we do histology, we can really quantify these. So we did these experiments, and we found that feeding the fish for a week with a high cholesterol diet is actually promoting steatosis. There is some injury, as you can see in there, hepatocyte injury with that arrowhead, but also macrovesicular steatosis with that little star that really, you can see pushes the nucleus of the hepatocytes to the side. And this is really a characteristic. And we see that this diet really induces these NASH phenotype. We also use several transgenic lines with markers for the nucleus and for the membrane of hepatocytes. And we have made a very deeply characterization using these lines in vivo with non-invasive intravital microscopy that allows us not to do histology, but simply look at the liver, and we can use the same animals over and over again to study progression. So that is an amazing characteristics of this model and these tools. What was really interesting to see is that feeding these fish with this high cholesterol diet for a week, we were actually, besides all the non-surprising findings that we had, like increased hepatocyte area, we actually found that this diet was starting to induce some kind of DNA damage that we could assess by the presence of micronuclei, just by really by using this H2B line that labels histone 2B. We could really see like how the presence of these little micronuclei that we quantified, and the diet is actually inducing, and we can use this as a surrogate for DNA damage. Besides that, we could also see that the consumption or this cholesterol surplus that leads to NASH in the zebrafish is inducing a lipotoxic environment as expected. We do see an increase on the caspase-3 in the hepatocytes. We also see an increase of proliferation, which makes sense. Cell death, proliferation in the liver are hand by hand in the development of the liver disease and even in malignancy. And also, it was very interesting for us to see that the high cholesterol diet in these NASH larvae is inducing changes in the vasculature in the fish. So we have a higher vessel density in the NASH larvae, which actually also mimics the vascular, hepatic vascular changes that we see in other animal models and in humans. But really, it was very interesting to us is to address the liver immune microenvironment in this NASH model. So using our amazing imaging skills and resources, we're able to, I hope you can appreciate how different the microenvironment is between a control and a zebrafish that is developing NASH. Or that has NASH already. You can see how these macrophages become really these fat cells crawling over the liver. These are like here in blue, crawling over the hepatocytes. You can really appreciate the vasculature changes here, like in these big holes in between, comparing like to the control. You can see how neutrophils are in contact with the endothelial cells, with hepatocytes and macrophages. So when we looked at these macrophages, that they looked also distant in the morphology, and when we quantified neutrophils, macrophages, and we also quantified for the number of these round morphology of macrophages that would suggest a more pro-inflammatory phenotype, what we found is that the cholesterol surplus was actually increasing the density of macrophages, and the ones that are rounded, as you can see in here, and also it was inducing infiltration of neutrophils, as it's a hallmark in human and murine NASH. Interestingly, we also found that the NASH larvae are actually, they have a decrease or reduction in the total T cells presence, which was actually quite surprising for us. But if you go to the literature, you can actually see how Tim Gretton's lab, for example, as there's actually evidence that an immunosuppression of the liver is actually related with NAFL, the associated HCC development. So I think our model is actually being able to recapitulate the immune microenvironment that we can find in the NASH, in the NAFLD progression. So importantly, these NASH larvae, they have an increased expression of inflammatory genes, oxidative stress, metabolic syndrome-associated genes, as we could measure by qPCR. And they all show systemic chronic inflammation. As you can see in here, neutrophils align very well in the hematopoietic tissues, kidney and color hematopoietic tissue, with macrophages here in green, resident macrophages all around. But when in our NASH larvae that were fed with a high cholesterol diet, we get this dispersion and this increase on the innate immune cells and the systemic chronic inflammation phenotype that is quantified in here, with 80% of the larvae at 1 week of feeding showing like a very severe phenotype of systemic chronic inflammation. So oxidative stress is a key trigger of hepatic cell damage and inflammation. With this in mind, Irene Pardo Sanchez, a PhD student at Victoriano Molero's lab, she has been using these mutant, these shuntine dehydrogenase mutant that she developed in the lab. And it was very interesting for them that by the time they found some papers that related XDH to non-alcoholic fatty liver disease. And this makes totally sense, since I just told you that oxidative stress is behind part of the development of the liver disease. This uric acid pathway that comes from the xanthine oxidase reductase activity and uric acid metabolism, it really produces large amounts of oxidative species. So we made this question. Irene came to make a short stay in our lab for 6 months. She's under mutants that we outcrossed with our lines. So we made this question of if an XDH deficiency could protect the zebrafish larvae from developing NASH. So the first thing she did, she did oral staining. So it's an easy way to address fat accumulation in the larvae and in the liver particularly. And what Irene found was that the mutants that were deficient in XDH, if you look at this condition in here, comparing to the high cholesterol diet wild type, has really a reduction of the steatosis, so the fat accumulation in the liver, suggesting that that the deficiency on xanthine dehydrogenase is actually helping or is ameliorating the disease or limiting the progression of the disease. So next, Irene also did some histology. It's a very good way for us to address steatosis and NASH scoring. So she really found that it was impressive, the mutants, the XDH mutants, when she fed with this high cholesterol diet. It was really impressive to see how these livers were healthy and nice without any steatosis and injury. And this is actually being scored at this moment. Because we expect that these XDH mutants actually had a lower oxidative stress in the liver. So Irene used this dihydroactinium probe that it can freely, enter the cell membrane and be oxidized by the cellular oxygen, by the cellular O2. So basically, she incubated the fish with this probe, and she took some pictures, and then she quantified the intensity in the liver. And what she found is actually that there is less ROS formation in the XDH mutants that were fed with high cholesterol diet comparing to the wild type that were fed with a high cholesterol diet, as you can see in here. So, but can the XDH deficiency reduce the neutrophil infiltration? We knew that was a characteristic on our NASH model and also a hallmark of the NASH disease. So Mariana, that some of you might know, Mariana Brandt do Amaral from Professor Niels Saraiva's lab at Unifesp. So she came to our lab, she got very interested in our NASH models, and she's very interested on the XDH mutants. So she's been doing some imaging, and the preliminary data seems to suggest that actually, yes, indeed, the XDH deficiency can also reduce the neutrophil infiltration. We could suggest that this ROS decrease, this decrease in the steatosis and the decrease on inflammation, it's actually part of the mechanism. XDH deficiency is being able to ameliorate the disease, or at least to somehow limit the progression of this disease in these mutants. But what pathways are being rescued by the XDH deficiency? To answer this question, we did some proteomics. So Irene, basically, she made these amazing, she took the livers from these tiny little larvae and she sent it to proteomics. We took about 20 per sample, and she was actually able to take more than almost 4,000 proteins in these samples, and she found about 170 different DEPs in their samples when she compared the control with the NASH. And it was very interesting for us that these NASH larvae, so we already knew that they have histological features that were conserved between with human and murine models of NASH. But it was actually very reassuring for us to show that there was a dysregulation of metabolic pathways and upregulation of several targets that has been previously associated with human NASH, such as, for example, ApoE-B, ferritina, AIF2-1A, and fibronectin-1, for example, are just some of the targets that we found in our proteomic analysis that are being upregulated. We also found that there were several pathways that are being downregulated, including FAO, cholesterol lipid biosynthesis, retinol metabolism, that actually retinol metabolism downregulation is very characteristic on NAFLD and NASH patients. When Irene compared the XDH deficient larvae with the wild type NASH larvae, it was very interesting to see rescuing on several of the pathways, metabolic regulated pathways, but also oxidative pathways. When we looked to the reactome pathways, we could see, like, for example, HSF1 activation is one of the top ones, as well as cytosolic sulfonation of small molecules and regulation of HSF1 mediated heat shock response. Searching for the literature, it is not new, actually, that the HSF1 activator ameliorates non-steatohepatitis, for example. We also found that activation of the heat shock response improves the biomarkers of NAFLD in patients with metabolic diseases. So it seems that this upregulation on these pathways could somehow be related to in the literature to an amelioration, or could justify how, or part of the mechanism that are behind how XDH deficiency can rescue NASH larvae. One interesting target is the sulfonation. That seems to be, really a key process involved in the liver detoxification. So salt enzymes or sulfotransferases, they are really important to metabolite hormones, catecholamines, xenobiotics, BPA, and other phthalates. And there's actually in the literature several papers that have been related these salts with NASH, with steatosis and cirrhosis development. So we have some pathways now that we can explore in the future. So in this first summary, I hope I convinced you that the XDH mutation actually protects the zebrafish from NASH and supports that XDH is involved in NAFLD disease progression, explaining this association that Mullero's lab had found before between XDH and the NAFLD in several clinical studies. So next, I'm going to talk a little bit, try to go quickly through a study that is already published, but it's actually been very important for us in the future to try. And the idea started for this study was really to try to understand what was the difference between a hepatocellular carcinoma microenvironment that has not been exposed to diet, to any kind of NAFLD, versus to a setting that we do expose the liver that is developing hepatocellular carcinoma to a diet that is rich in cholesterol. And this study was published in 2019 on Journal of Pathology. So liver cancer is actually the 6th leading cause of cancer mortality worldwide. It has a low survival rate. More than 700,000 new cases are diagnosed per year, with more than 600,000 deaths. So it's really a concerning issue worldwide. Only less than 25% are actually patients that are eligible for transplant. So it is really important for us that we really find the mechanisms that are behind malignancy, and also that sustain malignancy in HCC, and to be able to target them so that we can rescue the patients that cannot really go through transplant, or transplants are not actually, shouldn't be always the first option. There are really few available therapeutic options depending on of the type of HCC that we have, but hepatocellular carcinoma it accounts with about 75-90% of the cases, and fibromedullary hepatocellular carcinoma, it's another type of cancer, ultra rare, that I'm also going to talk a little bit later. So the liver cancer incidence in the US is actually, it's been increasing in the last 2 decades. It seems to have reached a plateau, but we still need to see the upcoming years. It is very curious that in 1999, this was the map, like how the incidence of liver cancer new cases, like in all US, and this is actually what is now happening, in 2019. What really happened in this, so we can really see how this incidence is increasing, and it really correlates with the high increase of NAFLD, and that is becoming the #1 cause to develop hepatocellular carcinoma. We know that there are many HCC risk factors. So for example, infections, toxic damage, metabolic syndrome, as I mentioned, and this all leads to this non-resolving inflammation, and this all leads to different immune microenvironments. So it's really important for us to have tools that we can study and that we can find drugs that can modulate the immune microenvironment depending on the setting or the etiology of the hepatocellular carcinoma. So in our lab, as I mentioned, we're interested in FLD. So we basically wanted to answer several questions, like how is NAFLD, for example, affecting the immune cells in the liver? How can we use drugs that are that have been suggested that they can rescue NAFL and NASH? Can they be used to decrease the diet, the enhanced tumorigenesis? What is the impact of this diet on the early malignancy, for example, and what are the immune cell players that are involved in the progression of NAFLD-associated hepatocellular carcinoma? So for that, we combine our high cholesterol diet model that I just talked to you a little bit, and we combine that with a model that was developed by Kimberly Evazon. These fish, they all the hepatocytes, they carry the potential to become a carcinoma. They are transformed. They are overexpressing constitutively active beta-catenin that is present. Beta-catenin and Wnt signaling pathways are present in about 50% of the HCC cases. These fish, they have hepatomegaly, so increased proliferation of hepatocytes. And about 6 months, you can see that about 80% of the fish, or really close 75% of the fish, already have HCC. So what is the impact of the diet on the NAFLD-associated HCC early in progression? So really early, using 2-week-old larvae, what is happening to the microenvironment? So we did, again, histological analysis. We found that HCC larvae that were fed with a high cholesterol diet, they have features of both NAFLD and HCC, such as lipid accumulation and glycogen accumulation, which is really great for us. Using our tools that I described earlier, these transgenic lines, we could address early malignancy characteristics that affect hepatocyte morphology to try to detect some of these early malignant features, such as, for example, cell size, nuclear circularity, nuclear area, and nuclear cytoplasmic ratio, all in vivo. And we did see that the high cholesterol diet induces all these malignancy features and enhances hepatocarcinogenesis. Interestingly, we really saw a huge amount of, using our surrogate for DNA damage, the presence of micronuclei in the hepatocytes. We saw that 60% of the larvae, they have these moderate to severe phenotype with a lot of micronuclei all around, comparing to the HCC alone. So really the diet, the cholesterol surplus, is pushing the system to get more damage. So when we looked at the vessel density, it's a very important feature in cancer to blood supply to the tumor, to help to proliferate and to sustain the tumor. So we really saw that the high cholesterol diet, again, is increasing this vessel density, which that could be probably related also why we're seeing an increase on tumorigenesis or hepatocarcinogenesis in these fish. So, but how does diet impact the HCC immune microenvironment? So again, we took our amazing ability to do imaging. We imaged larvae that they were developing, or they have the potential to develop hepatocellular carcinoma, versus the one that the transgenic larvae that they have the potential to develop hepatocellular carcinoma, but also they're being fed with this high cholesterol diet. And what we found was that these tumor microenvironments, again, the diet is completely changing them. In an HCC setting, we do have these macrophages that are more like with a phenotype that we believe to be more like M2-like, while in the presence of the high cholesterol diet, this is really inducing a very pro-inflammatory microenvironment with huge infiltration of neutrophils and these M1-like morphology on these macrophages. So we quantified all these again, as you can see. We do not have a change in the density of macrophages when we compared larvae with HCC versus HCC that were fed with high cholesterol diet. However, we do have almost a double amount of round macrophages. Almost all these macrophages seem to be displaying a more pro-inflammatory phenotype. And also we have an increase of the neutrophil density. When we took these TNF-alpha GFP transgenic fish that were outcrossed with a macrophage marker, we could really quantify the M1, M2, and we really found that the liver microenvironment on HCC larvae that have been exposed to high cholesterol diet, we have a higher increase on the M1-like macrophages, really supporting this idea that the high cholesterol diet is inducing macrophage polarization to a pro-inflammatory phenotype. So it was actually very interesting to see that again in this model of HCC with high cholesterol diet with NAFLD associated HCC, we also saw again that the presence of T cells is actually being reduced. But can drugs with the potential to treat NAFLD and NASH be used to reduce the diet-induced hepatocarcinogenesis that I just showed to you? So for that, we decided to use as a proof of concept metformin. So metformin is really an important drug for us to look, since it has been shown that is associated with a decrease of risk to develop HCC in patients with diabetes type 2. So we know that metformin has multiple targets, but one of them that is very well characterized is that targets complex 1 in the mitochondria. And we know that mitochondrial dysfunction and together as ER stress are involved also on the production of oxidative stress and also on the in the development of NAFLD. So it was important for us to really, besides this impact on mitochondria, metformin is also related to, it really targets also metabolic pathways. So we wanted to use this drug to try to see if we could rescue the phenotypes that we saw in our model. And indeed, we saw that. So using all these tools, we started to see that there was like a huge, when we compare HCC larvae with a high cholesterol diet with HCC high cholesterol diet that were treated with metformin, there was a huge reduction on the liver size it was also associated with a lower EDU staining of the hepatocytes. And then we using the transgenic larvae that I mentioned to you before, we addressed hepatocyte like early malignancy features like the hepatocyte area, nuclear circularity, nuclear cytoplasmic ratio, and micronuclei. And we were able to see that all these features were rescued when we compare the larvae that were treated with metformin versus the ones that were not. As you can see here, and the micronuclei were actually being able to rescue the diet effect, as you can see when you compare like with the HCC condition alone. So interestingly, we also found that metformin actually decreases the diet-enhanced progression, specifically NAFLD-associated fish, associated HCC fish, impacting also the vessel density. There is a reduction, that increase on the vessel density that we saw before in larvae, with HCC larvae exposed to the high cholesterol diet. Now we see a decrease. And interestingly, we also see amelioration of the steatosis in these fish. So when we look to the immune microenvironment, we are being able to reduce the TNF-alpha positive cells. As we know, most of the TNF-alpha positive cells in the liver are the ones that contribute with the TNF-alpha or macrophages. So we think that this is really like a decrease on that macrophage density. And we've been able to decrease lymphocytes. And surprisingly, when we reduce these, the infiltration of these immune cells and these pro-inflammatory immune cells, in this case, we are being able to restore the T cell levels to similar levels as we have seen like in the HCC fish. So as a summary, I just want to show you that that neutrophil macrophage infiltration also occurs in the HCC. The NASH-associated HCC, we have a change in this microenvironment to be that increases angiogenesis, neutrophil infiltration, it has a higher number of, it switches the polarization of macrophages to be M1-like macrophages, so TNF-alpha positive. There's a reduction on the T cells that is associated with increased steatosis, caspase-3, increased proliferation, and increased DNA damage. And when we treat these fish with metformin, we are being able to rescue all of these features that we have seen that the diet is pushing the system to increase hepatocarcinogenesis. And lastly, I'm just going to use the next 5 minutes to just tell you a little bit about these unique models that we developed also as I was a postdoc, that we've been using now in my lab to study this ultra rare type of cancer, the fibrolamellar carcinoma, which only accounts with about 1 to 2% cases of the liver cancer cases that are diagnosed every year. So fibrolamellar carcinoma strikes males and females alike, which is contrary to the HCC, that is usually more, the incidence is higher in males. Risk factors for fibrolamellar remain identified. So we really don't know what is causing the disease. However, it seems also that this disease affects primarily adolescents and young adults. Again, completely different from HCC that mostly affects the elderly because it takes really many, many years to develop malignancy. And currently, there are no effective treatment options besides surgery to these young patients. And most patients with fibrolamellar do not have underlying cirrhosis of the liver. And the initial diagnosis generally comes with symptoms that arise with advanced disease, unfortunately. So our work has been supported by Cancer Research Institute, but also Fibrolamellar Cancer Foundation, that are in a liver cancer model, in the liver cancer month. Please go to this foundation and see the work they've been developing and how they've been supporting science. And it's really important how they take care of their patients and how they try to promote these relationships with scientists, physicians, and patients. So there is fibrolamellars as one unique target compared to HCC that has many mutations usually associated to the malignancy in the tumor. There are many different types of mutations. In this type of cancer, usually is associated to one mutation, which is this fusion of the DNHAB1 with the PRKACA. And it's not detected in normal liver cells. It's only detected in the tumor cells. Importantly, this fusion retains the full kinase activity compared to the wild type protein kinase A. The replacement of the promoter of the protein kinase A with the DNHAB1 actually contributes to a higher basal transcription rate. And the PKA overexpression is associated with different types of cancer. PKA is known to regulate both the innate and the adaptive immune responses. So it became a very interesting cancer for us to study. So we published these in 2020 at TMM, so please take a look if you want to know a little bit more about the characterization process. But very quickly, I just want to show how this fusion is unique. That is the unique FLC molecular model, and how did we create this fish. So this fusion in the fish is in the chromosome 19. There are 2 isoforms per each one of them. There's a 400 kb. This is in humans, sorry. So in humans, it's on chromosome 19. These 2 genes are side by side. There's a 400 kb deletion that fuses the exon 1 of the DNHAB1, which is the HOC protein 40 gene, with the PRKACA gene, which is the protein kinase A, from exon 2 to 10th. In the fish, there are 2 isoforms per each one of these, and unfortunately, they are switched in the chromosome, so we could not just do a deletion to mimic the human disease. So we really decided to overexpress the fusion protein. We tried both, and we got founders for the alpha forms of both of these proteins. So we created this model with a marker in the liver and with the eye marker that allows us to screen. And we further, the first question we had was, can these fusion protein induce tumorigenesis in the zebrafish? And it does at the lower level, but it does. We were quite surprising with this. We were expected to be much more aggressive. There are several things that might be actually here playing a role. For example, we might need the beta form together with the alpha form, or we, for example, maybe using the hepatocyte promoter. This liver cancer type has been associated with cell stemness. So it might be that by overexpressing this fusion in the fully differentiated hepatocytes, we are probably lacking some part of the disease. So we are currently trying to do these overexpressing the fusion and even the human forms actually that are highly conserved with the zebrafish, ranging from 82 to 98%. We're trying to actually put these now under a more like hepatic progenitor cell marker to try to see if that will help push the system. But we do see that these fish are, they have increased proliferation. We can see that by the huge livers that we can see in adults. So hepatomegaly is definitely present in these fish. And actually when Francisco Juan, that is our postdoc in the lab that took over this project and won the CRI grant when he joined the lab, he really showed that these FLC larvae, by 2 weeks old, they have now this increased proliferation in the liver. But does the DNAJ PKC affect the innate immune cell infiltration to the liver, right? So we did some, again, we used our larvae, and we did see that there's an increase of macrophage as well as neutrophil density, which really which suggests to us that FLC livers, they have a pro-inflammatory microenvironment that helps to support the FLC proliferation. When we look to the macrophages, again, we also saw an increase on the M1-like macrophages at early stages of this disease. When Francisco Juan used, and these data that I showed it to you before was done like around 7 days, in larvae that were young, 7 days post-fertilization, right after the liver is becoming functional by 5 days. FJ also looked at T cells, so a 2-week-old fish. He really saw that there's also a decrease on the total density of T cells, and particularly the CD4 positive T cells. And this is not necessarily new. We found something very similar happening also in tumors from mice and from human samples that we've been analyzing using data sets available with timer algorithms. But which immune mechanism or which mechanisms are involved in FLC progression? So there's this paper with a murine model for FLC. There are currently only 3 models to study these disease animal models. So looking at this paper, we saw that there was an inflammasome complex upregulation when they analyzed the tumors versus non-tumor tissue, overexpressing this fusion. And FJ, when he looked, he saw really a huge amount of different inflammasome genes being upregulated. However, when he looked to some inflammasome components in human FLC tumors, comparing to the normal tissue, it didn't see. We are not being able to see that besides the pie card in here. So when we look to our fish, we do see that there is an increased caspase-1 activity using FLICA to detect that. So we wondered if neutrophils, for example, could be actually producing, or if inflammasome activation could be actually present in these neutrophils. So using these ASC-ASC-GFP larvae, we've been able to quantify pie card in the neutrophils, combining with the transgenic larvae that labels neutrophils, like in blue. So as you can see in here, the FLC fish that have an increased percentage of positive pie card fish, suggesting inflammasome activation. And when we treat these fish with the caspase-1 inhibitor, we see a reduction of these pie card staining or signal. Interestingly, FLC larvae that I have shown you before that have increased proliferation, when we use the caspase-1 inhibitor, we are being able to rescue that proliferation, which suggests that this inflammasome pathway could be related with increased proliferation that is seen on FLC and also in other murine models. When we did some proteomics of the FLC livers from the zebrafish larvae, what FJ found is that there are some actually interesting targets that came, not that many, but we did have some targets that came interesting. For example, the USP7 and the beta-catenin gene, together with CD42, are tightly related with proliferation. Very interestingly, these USP7, that is a deubiquitinating enzyme, erases ubiquitin and protects substrates from protein degradation. It's super interesting that if you go and search the literature, it has been associated with malignancy by affecting signaling pathways such as beta-catenin, NFKB, tumor suppressor proteins, for example. It avoids degradation of p53, avoids degradation of beta-catenin, and even some immune response and DNA damage proteins. It was very interesting that beta-catenin, a particular phosphorylation of beta-catenin, it's actually been seen to be a cancer-enhancing genome. It actually, beta-catenin cancer-enhancing genomic region axis is involved in the development of fibrolamellar carcinoma. And these USP7 and Wnt signaling, it seems to be very important in colorectal cancer to mediate the beta-catenin deubiquitination, avoiding degradation of beta-catenin, allowing it to go to the nucleus to promote proliferation and many other cancer malignancy events, such as, for example, probably this one in fibrolamellar. So I hope I could, show to you like that the overexpression of these fusion protein actually induces tumorigenesis in the fish. It induces innate immune cell infiltration to the liver and reduces the T cell presence. It increases the pro-inflammatory macrophages and activates caspase-1 in the liver. And we believe that our data suggests that inflammasome that is activated in neutrophils of the FLC, but also in the liver overall, might be involved in FLC malignancy, and the inhibition of caspase-1 actually reduces the proliferation of FLC. So I have a great team of students behind me that decided to join my lab and to help me to move forward my ideas, take my, be able to get that creativity, scientific creativity out there. For instance, the work that I showed today, fibrolamellar work has been developed by Francisco Juan. He's a CRI fellow and fibrolamellar carcinoma fellow, as I mentioned. He's also being supported by B+ Foundation. Mariana in here, she's also been joining the lab to study NASH, and she helps with the XDH. And Irene, that she's not in here, but you could see her nice face. She was actually very important for us to show how our NASH models actually have conserved features with human and murine NASH. I also want to thank to all the collaborators, Victor, my amazing PhD mentor, Neil Saraiva, Mariana's mentor in Brazil, Teresa Bowman, Simone Sidoli for the proteomics, and all the people that are helping us to develop the reference diet, and all the Einstein facilities, core facility services that have helped us, and all the funding that we are getting to help us to move science forward. Thank you so much. Now I can answer your questions.
Thank you very much, Professor Sophia, for the amazing presentation. I'm sure everybody enjoyed it. Okay. Now, Professor Sophia is going to answer some questions that were asked during the presentation. The first one of them is, do you have any tips to facilitate histological processing zebrafish larvae?
Sorry, could you repeat? It's raining a lot outside.
Oh, no problem. So, do you have any tips to facilitate histological processing in zebrafish larvae?
Yes. So it took us a while to do it. So the first time we took it to our zoology services, I remember like in medicine, they couldn't find them, right? So what we've been doing, and now it's a very straightforward protocol to us, we first put them in these micro cassettes. So we put, we actually first, put them in the formalin, and then we put them in agarose. So we basically align, like, I don't suggest to go over 3, 4 in a line, and then you put like 1% agarose on the top of them, so that it squishes them to be all aligned. And then you cut like a little square around, and you transfer that to the micro dissection cassette. And that has been the best way for us to get the livers, like the larvae, aligned, and we can do like really nice histology of the larvae. Yes, and we've been doing these in multiple, from 7 days up to a month in adults, and it's been working really nicely. Yeah.
All right, nice. And there is another one too. Let's see. So, have you ever studied the production of specific immunoglobulin by liver tumor cells and the modulation of the liver immune response?
No, we didn't. And this work is actually right now kind of like in standby, trying to find people to come and do it. But yes, there's a lot for us to do, and it would be interesting to try to see how, but there are also a lot of people working on HEC. Kimberly Evazon that developed this amazing lab, the tool, she keeps using it, and other ones. Labs in Singapore also use it a lot. Wolfram Gosling in Boston. So yeah, but we need to move forward. We are the only ones doing NAFL associated HEC, but we also are very eager to see if the new NAFLD models that we're trying to develop, if they will develop spontaneous HEC, so that we don't need to combine necessarily with the beta-catenin. Of course, it's going to take probably many, many years. And this beta-catenin model is just an amazing tool for us to address early malignancy features at really early in the larvae stages. Otherwise, we really need to wait a lot of time.
All right, that's nice. Okay, so thank you again, Professor Sophia, for all your answers and for your presentation. Okay, it was a pleasure to have you with us. And this concludes today's webinar. Okay, so thank you all for attending. Hope you enjoyed, and we hope to have you back here tomorrow live on our YouTube channel, the Institute of Biomedical Sciences YouTube channel called ICB-USP. Okay, so have a nice night and see you tomorrow, guys.
Thank you.
My name is Mark Francis. I'm the founder of Aquaneering. For the last 20 years, Aquaneering has built a team of dedicated professionals to support your research. Our mission is simple: provide the highest performance zebrafish housing at a reasonable cost. Our team is here to allow you to focus on your research.
We at Alesco work daily to create solutions and equipment for the scientific community, contributing to the advancement of Latin America biomedical research. Our mission is greater than just providing equipment. It is to provide security with a quality service and mainly with enormous respect and attention. For this reason, we invest in high technology and seek to maintain long-lasting relationships. Our commitment is to be a reliable partner that understands the needs, the reality, and the conditions of each customer to always offer the best solution. We understand the benefits of scientific research for humanity, and this encourages us. If today we have a better quality of life, greater longevity, if we beat cancer or use a medicine for a headache, it is because the advance of biomedical research allows us to. We trust in the work of researchers, in science, and the scientific community, and we pride ourselves on doing our part. Just like you, we are passionate. Science is what moves us, because for Alesco, research is for life.
Welcome everyone to FST's presentation. We appreciate you taking the time today and look forward to helping you find the right tools for the right application and your endless discoveries and research. Find Science Tools has been in business for over 45 years, providing scientists, researchers, and other life science professionals with high-quality surgical instruments utilizing German steel and German craftsmanship with a secondary quality control inspection in our Heidelberg office. As we say, we only carry the best and offer a multitude of fine microsurgical instruments like spring scissors, forceps and rongeurs, but also a variety of options for wound closures and animal identification. In addition to our 3 FST offices, we have over 50 distributors worldwide. While the majority of the audience is based in the USA, please note that we can ship anywhere in the world, and any products not listed in our physical catalog or online at findscience.com can be sourced as a special order, and we will work with you to get you what you need. Before we start, for those who are unfamiliar with FST, let me go over our core values we hold ourselves to and strive to provide to our customers. Quality. Impeccable product quality is what differentiates FST from the others. Secondly, our customer support. We strive for 100% customer satisfaction. Lastly, our QC department in Germany upholds our manufacturers to the standards FST and our customers expect with every instrument.